We're just gonna suggest what to do.
We're good now.
Oh my God, it's you and Billy.
Yeah.
We're not getting pressure.
Yeah, that is.
It feels funny using a microphone. But we may have people watching on Zoom. So thank you all very much for coming. It's actually very exciting to have a Interfaces Symposium in person. Hopefully more people will join us during the day. Also, I wanted to use this opportunity to wish everyone a very happy International Women's Day. And to thank all of the organizers of the symposium today. So we have Emma and Marcus and Kristen and Sean and Joseph and Stephanie. And I know it's quite a lot of work and oh, sorry, I missed, oh, Nate, sorry. And it's a lot of work to put these things together, and it's sort of also a lot of uncertainty right now. So it feels like on the one hand, it feels like, of course, we should be doing this. And on the other hand, it feels like, wait, I'm standing in front of people talking, and what do I do? Where's the camera? So I really appreciate you putting together this program, and it's actually a very exciting program, and which I'll tell you about. So our keynote speaker is Professor Comavez, and Dr. Comavez has been probably the single most important faculty member in the success of this training program. She's very experienced in running NIH training grads, and she's really understands what students go through and really shows a great deal of interest in their progress. And when we have steering committee meetings and we talk about students, somehow she seems to know who all of them are, which is amazing because we actually have 11 different graduate programs in the Interfaces program. So we'll be hearing from Professor Converse today about her research. She's in the Department of Chemistry and Biochemistry, and we're really looking forward to that. Then we'll have one of our newest faculty members on campus, Dr. Stephanie Lindsay.
Actually, in the afternoon. She's in the afternoon.
In the afternoon, right? Okay, so this.
So that one.
Okay, so the timing of the schedule isn't right, but Dr. Lindsay has a great deal of experience in the biomechanics and mechanobiology of development, and she trained in Washington University, and then in Cornell, and then at Stanford, and actually there she had a UCSD connection because she trained with Professor Alison Marsden, who used to be here, and the MAE department was very fortunate to recruit her last year, and so we're really excited to have Professor Lindsay join us and to join the program. Then we'll have the lightning talks, and I don't quite know how. I guess it's going to be something of a hybrid session. We'll see how it goes. But I attended the lightning talks we've done here before have been really good, and I attended one online that was hosted by a foundation the other day that one of my students participated in. And I was like, this is so cool. It's actually a great format, and it's actually a very good format for Zoom. So not that we should be planning Zoom things, we should be hoping to see the end of Zoom, but I do think the lightning talk format is a great one, and we've used it successfully here before. So and then we'll have lunch, and hopefully it'll be just as nice then as it is now, and we'll get some nice photos. And we have some alumni from industry and other careers here. Actually, we already have a couple of program alumni here with us, Ernesto and Kim. So thank you very much for coming. And we'll have some more of our alumni here to talk about their sort of career paths, and really looking forward to seeing them. We also have another panel on academia, and then one of our last speaker, faculty speaker, is also a new faculty member, so Professor Ben Smarr from bioengineering, who does data sciences and particularly studies circadian biology and uses big data sets to understand the effects of circadian rhythms on human health and performance. And if you recognize the name Sparse, you may remember that the founding director of what's now the Qualcomm Institute, was Cal 82, is up there, Larry Sparse. He's actually also the guy who, when he was at NCSA, his lab wrote the first web browser, the Mosaic web browser. That's Ben's dad. So, we're really looking forward to hearing from Ben. And Ben's also expressed interest in offering the course that we've talked about that during some of our retreats in the past on data sciences. And just when we were ready to sort of ramp all that up, the pandemic came, and we all got worried about other things. And so it'll be very exciting to hear his ideas on how we could have a course for interfaces students on data sciences for multiscale biology. Okay, so most of you probably know a fair bit about the program, but we have at least one first-year student here. And actually, we've been going long enough that probably none of you know the full story. So this program actually started back in 2005 when the Howard Hughes Medical Institute announced something they were calling the Interfaces graduate programs, training programs. And it was a 3-year grant, not so much to be a training grant, but to develop a training program, an interdisciplinary training program. And the name Interfaces actually predated that. They borrowed that name from an earlier program that the Burroughs Wellcome Foundation had used. And in fact, UCSD had one of those awards, was called La Jolla Interfaces and Science. And so the interfaces, they really liked that interfaces concept of bringing physical sciences and biological sciences, mathematical sciences, computational sciences, engineering sciences together, and deliberately doing training at the interfaces. So Howard Hughes Medical Institute picked that up. Now, by 2005, UC San Diego already knew how to do interdisciplinary. We had one of the first and most successful bioengineering graduate programs in the world, one of the first and most successful neurosciences program, a chemistry and biochemistry program. So the idea of doing interdisciplinary science wasn't new to us. But we're like, you can't sit on your laurels, you've got to go to the next level. And so we said, there's a lot more scope for interaction than simply engineering with medicine or chemistry with the School of Pharmacy. And so we really sort of designed this program as interdisciplinary graduate research, the next generation. And I think we've really sort of lived up to that vision. We got one of those grants. We didn't use it so much for development. We actually immediately used most of the money to support trainees and created the training program. And then the idea all along was that the NIH would sort of take over and have a longer term support through the T32 training grants. And so we were one of the successful programs in getting that, and we've managed to renew it ever since. Thanks in large part to the expertise of people like Dr. Comaves and the hard work of the staff like Irene and Kelly. One of the things we had to do to help us get that was actually to formalize the program. And the way we formalized it was by making it an interdisciplinary degree specialization. So as you mostly will know, when you join the specialization, you become eligible to be a trainee on the training grant. But everyone who, whether they're on the training grad or not, becomes a member of the specialization. And what that means is when you graduate, by meeting the specialization requirements, you graduate with your home degree, whether it's in mechanical and aerospace engineering or neurosciences or biological sciences or biomedical sciences, and a specialization in multiscale biology. So that's the formality, and that was in 2009. And since we're now in our 13th year of this training grant, and those are all the participating programs have now expanded to include nanoengineering and material sciences. And we currently have over 30 students and more than 23, I think more than 25 graduates. So anyway, blah, 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 okay. We'll tell you about you either already know because you're in the program, or you'll find out in the spring about the details of the specialization. But essentially what we designed it to do is that the curriculum is lab-based, and it involves technologies at different scales of biological organization. And so here are the courses. And from supramolecular complex characterization, which is a course that Dr. Cumens and Gazemian teach, and mass spectrometry, all the way through to whole body magnetic resonance imaging. And trainees take 3 of these classes, and we try and construct it in such a way that by cross-listing courses and other things, so that at least some of them will actually count towards your degree of progress anyway. So maybe you end up doing an extra course or 2, but these are also hands-on courses that will help you in your research, and you take your own samples of data along to. And we'll be talking, as I said, about adding a data sciences course, and with Dr. Peter Wong leaving, we'll also be talking with Dr. Schoenberg about creating a next generation imaging light microscopy class. Dr. Schoenberg is an authority in lattice light sheet microscopy, and this is a super cool method for watching the dynamics of live cells, or even in tissues, fairly deep into the tissue, and surprisingly high resolution and frame rates. Okay, this is all sausage making of admission into the program, which again we'll cover more about in the spring. And I think that leaves us just enough time for you to introduce Professor Cumens. And again, thank you all for coming. Thank you all organizers. Thank you to Kelly and Irene for all your work. And if no one else shows up, we're going to be eating a lot today.
Again, yeah, thank you all for coming. As Dr. McCulloch said, our first keynote speaker is Dr. Cumens, who is the current chair of chemistry and biochemistry, and has been an integral part of the faculty that's part of interfaces, and really, made interfaces what it is. I know that she's been a pioneer in HDX MS, and I think that will be one of the topics that we'll cover in her keynote. And so if you will all welcome Dr. Cumens to be our first keynote speaker. Yeah, I think I should.
Made a bad mistake and forgot to bring my adapter.
USB-C.
No.
Oh, wait.
This is like a regular Mac adapter.
Oh, you're right. I do have an adapter for you.
Oh, you do? Oh, that's great. It's really bad after being in Zoom for so long. There's things that I haven't given a talk in public. Yeah.
The other option actually is if you want to on a thumb drive. Yeah. Yeah.
Oh, and then I need the Zoom as well.
That would be possible. Record your screen.
I have it. Didn't put it in. This one was listening to him.
Apple.
685.
Yeah.
The other option is.
Oh, it did. It did. Yeah. There we are.
All right, so now you're gonna have to help me. Oh, I'm.
All right. I just need to turn off the sound on my computer.
I'm so sorry. Sorry about all this.
No, that's perfect. But somehow it's still sounding here. Okay,
I think so.
can you guys hear me?
Yes.
All right. It's gonna be interesting. Is it okay if I just, oh no, it's not okay. I'm trying to figure out a pointer. That's the last thing I need to do. There we go. Beautiful. Okay. Well, I can't tell you how much of a pleasure it is for me to be here today. I've been at UCSD since 1990. That's why my hair is gray. And I was involved with the very, very, very first La Jolla Interfaces in Science program way, way, way back in the day before Howard Hughes. Well, it was Burroughs Wellcome, and then it was Howard Hughes, and then now it's an official T32 program. But, you know, we were one of the first universities in, I think, the world to get this idea that it could be fun for grad students to have two thesis advisors in different and complementary fields. And part of that was because I had really been enjoying collaborating with people who I didn't understand what they were saying, right? And this was the idea that different fields speak different languages. And unless you're exposed to those different languages that people in physics speak or people in engineering speak, you can't really work together with people, right? And so that was also the idea of these lab courses, was to immerse you in, you know, tissue speak or imaging speak, and, you know, have you have real experiences with doing things that are outside of your area of expertise. And that, I want to encourage you all, when you do something that's outside your area of expertise, you become more creative. And that's what's happened with, you know, the trainees from this program over the years. You know, many of them have gone on to become really famous scientists because their creative juices got working at an early age, if you will, right? So that's the idea. But anyway, today I'm going to tell you about my own research, which hopefully you will see ways that we've sort of put things together to think about how proteins move. And so that's what I'm going to tell you about today is the biophysical insights that we've had into protein dynamics in signaling proteins. And I don't know if I'm going to get through everything that I'm going to talk about today. It's been so long since I've given a talk, I don't even know. I couldn't even think about what to talk about. But anyway, Sean mentioned that he was interested in having me talk a little bit about some of the insights we've had in serine proteases. And then I want to tell you some of our latest work in NF-kappa B. And then if we have time at the end, I'll tell you about this giant ubiquitin E3 ligase. Let's see if we can get that far. All right. So physicists don't think of proteins as fixed molecules. Physicists think of proteins as being splayed out on an energy diagram that obeys Boltzmann's equation. So in Boltzmann, we know that we have different ground state energies for slightly different conformations of a protein. And those ground state energies can interconvert with each other over energy barriers. And the ground state population differences are defined by the energy differences. So there's like a higher percentage of this one than this one, and then a higher percentage of this one than this one. And these are related by Boltzmann's equation of the difference in the energy, okay? And so when I think about proteins, I actually was trained, believe it or not, as an organic chemist. But then I became a protein biochemist because in my mind, proteins are just large organic molecules. And the way that you learn that organic molecules move is the same way that I think about how proteins move. It's just that there's a whole lot more energy states involved because we don't just have one, you know, bond rotating, making the gauche and the syn or anti or whatever, but you know, we've got that times 10,000 or something like that. And now, even more, I think it's pretty cool that when proteins bind with each other, they actually change the way that they move within themselves and with respect to their binding partners. And so that's what my lab has focused on over the years. And so it turns out that from Boltzmann, we know that those energy barriers between one state and another are pretty low. And so proteins don't exist. I mean, even though you see crystal structures of proteins, they don't exist like that in solution. They exist in this ensemble of interconverting states that are all sort of similar in energy. And they're moving on a very wide range of timescales. And so I like this slide because it just kind of shows you, you might think about femtoseconds. That's like a time step in a molecular dynamics simulation. And that's, sort of bond vibrations. And then we have, the nanosecond to microsecond regime. This is a very interesting regime of motion. It's the rate at which proteins diffuse in solution. It's also the rate at which a loop will go like this in a protein. Okay, and then picoseconds to nanoseconds are side chain rotations. I always tell people one of the sort of eureka moments for me was, I don't know if you've ever looked at a crystal structure of a protein and you see this beautiful aromatic ring, and it's got protein packed on this side and protein packed on this side. And you think, oh, it's there. It knows exactly where it's supposed to be. But then the NMR spectroscopist measured this aromatic ring and it's going flip, flip, flip. And I've slowed this time scale down by 10,000. It's flipping at the nanosecond. Every nanosecond it's flipping. Okay, and so you see it always like this. Right? It's really, the snapshot just took it because it's not like this as often as it's either like this or like this, right? So when you take a snapshot, you're going to see it packed, but that's not how it is in solution. And then you imagine that happening over the whole protein, and it's just fun to think about what these things can do, right? So, okay, and then now lately, my lab has gotten into even longer time regime motions, milliseconds and seconds, so I'll show you just a tiny little bit of NMR CPMG data, hydrogen deuterium exchange data, and now we're even pushing this arrow further out with single molecule FRET. And I want to try to tell you about all of that today. Let's see if I get there. Okay, so so one of my friends who is a physicist is a guy named Peter Wollenis. He's actually the guy who first said, you all think it was Ken Dill, but it wasn't. He's the guy who first said that proteins fold along a funneled landscape. I'm sure you all have seen the picture of the protein folding funnel in your textbooks. And Peter never gets credit for that. He did the physics behind it. This is one of the things that I will tell you happens very often in interdisciplinary science is that someone will take something that you did and explain it to biologists. And then the person who explains it to the biologist gets the credit for the idea because the biologist can't understand a word Peter Wollenis said, right? And it took me a whole year of having lunch with the man every single week to understand anything he was saying. But then when you understand what somebody outside your field is saying, you're like, oh my God, this makes so much sense. So what Peter and I did together was this idea of localizing frustration. And, in graduate school, you can relate to frustration, right? But frustration is a polymer physics term, okay? And what it means, it's really a really cool concept. So I don't know if you've ever thought about this, but glass is a collapsed polymer, okay? But the thing is that the monomer units of the polymer are all the same. So when a glass molecule collapses, it could collapse like this, or it could collapse like this, or it could collapse like this with all different things around it. But all of those energies of all those interactions are the same. So the glass can't figure out, do I be like this, or do I be like this, or do I be like that? Because it all kind of is, the same energy. But this is what Peter said about proteins. He said when proteins collapse, they have 20 amino acids and they've evolved to fold. And so the energies of the contacts between the side chains are actually, there are some that are very preferred. And so he called that minimally frustrated. Well, who knows what that means, right? But what it means is that that amino acid wants to be making the interactions that it's making. Those are energetically preferred. And the way we measured this was just with a simple energy function. You take this amino acid and you say, what if your neighbor was anybody else? Do you care? And some amino acids really care because they're minimally frustrated. And other amino acids say, you know what, can I please interact with that other guy? I like him much better than the one that's next to me right now, right? And those are the frustrated ones. They don't want to be where they are. And it turns out, so this is a picture of thrombin, and the red lines are all the frustrated contacts, and the green lines are all the minimally frustrated contacts. And you see this for every protein structure, that the core of the protein is minimally frustrated, and you see big patches of red lines on the surface, and those are the binding sites. It totally makes sense, right? But it makes sense at a deeper level when you understand that it all has to do with the energies of the contacts of the amino acids and who they want to be interacting with. And the really cool thing, so I did this this graph for a paper that was in honor of Peter. I can't remember what birthday it was that he had, but anyway. So then we measured the order parameters in thrombin. Okay, how do every NH group in the protein move? Are they moving? Are they not moving? Right. So the lower the order parameter, the more that part of the protein is moving. Okay. So I plotted these order parameters on the top. Here's a disordered region. Here's another disordered region. Here's a really disordered loop. Here's some more disorder, right? And look how it perfectly correlates with the frustration index. And that totally makes sense too, right? Because, you know, if you're very happy where you are, making all the interactions that you're making, why would you move? But if you're frustrated and you really want to be interacting with somebody else, then you're going to try to move. And so this is the fundamentals of how proteins are built. They're built to have minimally frustrated regions and then frustrated regions that move. And if a protein is completely designed to be minimally frustrated, it's not ever going to do anything. Proteins have to have frustrated regions in order to work. Okay. So then, right. So then we were thinking about, so my lab has been doing amide hydrogen deuterium exchange for a really long time. You take a protein, you soak it in deuterated buffer, and you watch the NH groups exchange with the heavier hydrogen isotope, okay? Now, the problem with that experiment is we don't know what it's telling us. Because we have all these amides, you know, and they're exchanging at different rates. It's an amazingly powerful experiment. It was developed in the 1950s, actually, before we even knew about deuterium. Do you guys know who invented or who discovered deuterium? There's a building on campus named after him. Yes. Harold Urey, yeah. He was one of the founding members of our chemistry department. Before deuterium, they knew about tritium. So in the early days, they used to do this experiment with tons of radioactivity. I have a thing about not doing anything with radioactivity because I'm so old that, you know, I was from that era where most biochemistry was done with radioactivity. And I had a colleague, he's a famous scientist now at Harvard, but we were doing some tritium experiments and he decided that instead of using a syringe and a no air approach like he was supposed to do, he just opened the hood, took a Pasteur pipette, took the tritium that he needed and, you know, did his reaction. And then he discovered that he was peeing out 10,000 counts per minute of radioactivity from his urine, right? So the solution was we had to take him out every night and get him drunk until he peed all the tritium out of his system. Then he freaked out because he couldn't walk a straight line anymore and he thought he had done some neuro damage. But it turned out that this is like a defect in his family that nobody in his family can walk a straight line. So in the end, he's fine, and he's a professor at Harvard, right? But okay, that's a sidelight. Okay, back to this deuterium exchange. So so everyone has always understood deuterium exchange in this sort of simplified equation where you have a very stably folded protein, they call it the ground state, and then this NH group is in a hydrogen bond, right, in the ground state. But then if that protein breathes or opens somehow, that NH group can become exposed, and then it can exchange with the deuterium, okay? And the tricky part is that for most amides in your protein, this equilibrium is very much favored towards closing. So the NH group is going to go, boop, boop, right? It's going to stay closed almost all the time, but every once in a great while, it's going to open. And then the question is, when it's that split second open, does it have a chance to exchange before it closes again, right? But it often doesn't. And so what you're really measuring in amide exchange is this equilibrium of, how often does this thing open versus most of the time when it's closed. So it turned out that we could measure this excursion to this excited state by NMR. And we did this in thrombin, and we measured the K open was 170 per second and the K closed is 1700 per second. And we took these numbers and we put them into the amide exchange equation. And we came up with this, this rate of exchange. Now this is why the reason I'm telling this is because people get so confused about this. But the rate of exchange doesn't tell you how the protein moves. It's like an after effect measurement because of this equilibrium. All right, so we are opening at 170 per second, but our amides are exchanging about 1 per minute. Got to wrap your head around this to figure this out, right? But anyway, so this kind of makes life nice for us because we can measure amide exchange over the minutes time scale. And what it's actually reporting on is these millisecond timescale motions. And these millisecond timescale motions, if you remember back to my little graph, are the really important ones because those are like, domains shifting with each other, loops opening and things like that. And that's what proteins do for their function. All right, let's keep going. Otherwise, if I tell you too many anecdotes, we'll never get out of here. All right. So I want to tell you about NF-kappa B and maybe ubiquitin ligases. I should have looked at my time. Okay, we're fine. All right. So so NF-kappa B, we've been studying this for a while now. It's a stress response transcription factor. And basically it sits in the cytoplasm of cells until a scary signal comes from the outside. And then the cell goes, oh my God, stress. And it turns on NF-kappa B. And we aren't going to worry about how right now. And the NF-kappa B goes into the nucleus and turns on all these different genes. And actually, that's one of the theories of, or one of the focuses of my research is, how do proteins turn on and off? If you think about like serine proteases, I mean, you have tons of serine proteases in your body and none of them are working right now. You have multiple systems for keeping those things off because otherwise you would self-digest. That would be a really great horror movie, wouldn't it? And it's the same thing with NF-kappa B. You absolutely want this transcription factor to be off all the time because otherwise you would have cell proliferation, which is cancer, inflammation, immune response. Okay, so this is one of the things. It's like really interesting to me how our proteins control their own activities. So I don't really want to go into this. NF-kappa B is a whole family of proteins. I'm only going to tell you about one of them today, but just notice how all these different family members bind DNA in the same place. The protein kind of hugs the DNA. You see it? They're all doing that. They're all hugging this piece of DNA. So of course, everybody thinks that that's how they always look, right? They're sitting there with the DNA in the hole in the middle of them. Okay. And then the way they actually work, the way they're actually kept off, I guess is a better way to say it, is that there are these inhibitor proteins. This is how I got into this thing, because I thought these inhibitor proteins were really interesting looking things. Actually, this is how I roped Peter Wolonis into working with me. I taped the picture of this protein onto the door of his office, and I said, I bet your protein folding algorithms can't predict this one. And he took that as a challenge, and we started working together. So NF-kappa B is really cool because, like I said, it's all in the cytoplasm. Oh, this is not good enough. All right, well, darn, can I undo the slideshow and just show you this video? Screen sharing has stopped. Well, only the people who are present get to see the video, is that what it is? All right, watch the nuclei are all green right now, right? Okay, so see how, so the orangish color is the NF-kappa B. So it starts out in the cytoplasm, and then upon stimulation, it all goes into the nucleus, but then it all comes back out really fast. Now this is sped up a little bit, but it's really fast. Okay, so now let me go back to, now do I have to screen share again? Yeah, let me see how I do this. And slideshow, screen share. Yeah, I don't want to really be too discriminatory. Is it this one that I showed? Oh, no, this one. That's what you did the last time at work. I watched that. I'm trying to learn in my old age. Are we good?
It's actually on the actually.
No, you're good. I'm glad you're here for me. There's so many windows to pick from. We could be here for a little while. Okay. Yes, thank you. Okay, and so if you measure the NF-kappa B, what you see is that it goes into the nucleus, out of the nucleus, into the nucleus, out of the nucleus. It oscillates like that, right? And I won't get into all the details, but that oscillation depends on one inhibitor. It's called I-kappa B alpha. And we figured out exactly what I-kappa B alpha is doing. Oh, look, it would have gone. Okay, well, now you people on Zoom can see it. All right. Okay, so here's the story that what's happening is this inhibitor, I-kappa B alpha, is very, very tightly bound to the NF-kappa B in the cytoplasm. And then when you get a stress signal, the I-kappa B is phosphorylated, ubiquitinated, and degraded. And when the I-kappa B gets degraded, then the NF-kappa B is free to nuclear localize into the nucleus. And that's when it starts turning on genes. And one of the genes that it turns on is the gene of I-kappa B alpha, which, this is the fun part. It's made out here in the cytoplasm, and somehow it still manages to get back into the nucleus and turn this whole system on. Okay, and the way it does this is by a process that we are now calling molecular stripping. So by hydrogen deuterium exchange, this inhibitor, I-kappa B alpha, has a really interesting property. Even though it looks folded in the crystal structure of the NF-kappa B, I-kappa B complex, the free protein, these last two repeats, are actually completely exchanging their imides. And then when it's bound to the NF-kappa B, they're not exchanging anymore. So this is a case where this protein is folding when it binds to its binding partner. Okay, that's why its imides are not exchanging anymore at the end. This is just how we do the deuterium exchange experiment, but I mean, I don't think I really have to go through this. You soak the protein in D2O for a few minutes, you quench, you get peptides, you shoot them onto your mass spectrometer, and you measure how much deuterium they took up. And it's neat, right? Because we call these ones control peptides, right? There are regions of the protein that don't change. I don't know if you can see the red dots are right underneath the blue dots here. So there are parts of the protein that don't change at all, and then there are parts of the protein that have these massive changes. And that's what's so powerful about amide exchange. So, and we can map the whole protein this way, and we can see that, the middle isn't changing very much, but the ends are changing a lot. And, we can color the protein picture and show you that, this is exchanging a lot in the free protein, and it looks all folded in the bound protein. And we did this actually, I don't know if there are any computational people here today, but I thought this was a really nice demonstration of one thing that amide exchange tells you. So, so these open circles here are the last two anchor repeats in this anchor and repeat protein, I kept the alpha. And in the free protein, you can do a solvent accessible surface area calculation. Maybe some of you guys have done this. You take your protein structure and you roll a water molecule all over it, and you say, how much should this part exchange? Okay. And then we correlated that with the deuteriums incorporated. And in the free protein, all the peptides from this 5th and 6th repeats are exchanging way more deuteriums than they're predicted to exchange according to the structure. Okay, but in the bound protein, they fall right on the line. So that says that this protein is folded in the bound state and not folded or not folded well in the free state. But one of the things that I've really started to think about is, what does it mean to be not folded? Is it spaghetti? Or is it this molten globule that's got helices, but they're just kind of not knowing where they are, So that's why we ended up going ahead and doing single molecule FRET. So the idea of these single molecule FRET studies is that you put a donor dye and an acceptor dye on your protein. And then what's going to happen is you shine a laser on the donor dye. And if it's close enough to the acceptor dye, it's going to transfer energy. Okay. And then the acceptor dye is the one that's going to fluoresce. But if they're farther away from each other, then even though you're shining laser light on the donor, it's too far away to transfer energy to the acceptor. And so the donor still fluoresces and the acceptor doesn't fluoresce much. And it turns out that this FRET depends on 1/r to the 6th. So very small distance changes show you big differences in FRET. So this is the way we set up the experiment. We set it up in such a way that we could watch this molecule for a long period of time because we attached it by an antibody to a histag on the microscope surface. Okay, and I think this is just the coolest thing ever. What we saw were traces that looked like this. Okay, now what's happening here? We're shining light on the green dye, and it's transferring all its energy to the red dye. So this means the dyes are close together. But then over time in seconds, look at this. These dyes are moving. They're close together at the beginning of this trace, and then they go farther apart, and then they kind of go like this, and then they go close together again. They hang out there for a while, and then they go really far apart. That's right here, right? And then they go close together again. So this is actually, I think, the first demonstration of a protein moving that we could watch the ends of it. I mean, now this protein looks like a slinky, right? So you can think, okay, it's, it's behaving like a slinky. And we're watching it in time over the order of seconds. This is a really slow, in what you normally think about as timescales of motion. And then we can put the dyes in two different places and show that, the 5th repeat doesn't do this. It's only the 6th repeat that does this at room temperature. And we did a whole bunch of controls to make sure this was right. We had this stabilizing mutant that at 25 degrees didn't do the slinking at all. But if we turned the temperature up to 37, then it started to lose grip. This is really a fun set of experiments. But anyway, okay, so, and we know that biologically, this free protein has a very short half-life in cells. if it does not find an NF-kappa B to bind to, it gets degraded really fast. All right, and so we showed, this is in collaboration with Alex Hoffman's lab. Okay, this is the degradation, right? Watch this, wild type I-kappa B alpha. There it is at 0 time, and by 10 minutes, it's already gone. Okay. And then we made the stabilizing mutant that hangs around for a bit longer. But what was really, really cool, Alex's lab did this. He took the 6th repeat and stuck it onto GFP. Now this is, right, so this is the real definition of something called a degron is, can I stick this on GFP and the GFP degrades? Okay, and that's what he did. Look at this. Here's beautiful GFP, right? But then when you stick the C terminus of I-kappa B alpha on the end of GFP, boom, it's gone, just like the I-kappa B alpha was. So, okay, so, and then of course the stabilizing mutant lasts longer, but so this is really neat, right? Now, let me see if I can go backwards. Yeah, so one of the ways that this system is under really exquisite control is that this complex is super stable in cells. If you don't get a kinase reaction or an E3 ligase reaction, you could sit there for days and this complex would not go anywhere. But if you get those signals, the inhibitor gets degraded, the NF-kappa B goes in, it starts making I-kappa B alpha. But the I-kappa B alpha that it makes is very unstable because you don't want free I-kappa B alpha around, right? Why don't you want any free I-kappa B alpha around? Because if you get a stress signal, you don't want free I-kappa B alpha to be sopping up this stuff because then it can't do its job to say stress, right? So the system is all set up in such a way by the degradation of the inhibitor, the time scale of the degradation of the inhibitor, and that's all controlled by the fact that the C-terminal part is not folded in the free and is folded in the bound. Okay. Let's skip this part. Yeah. Well, okay. Let me tell you what we figured out. Maybe I should not skip. So what we figured out is that I-kappa B alpha causes NF-kappa B to dissociate from the DNA. So not only does the I-kappa B alpha have a different degradation in the free and the bound, when the I-kappa B alpha is newly synthesized, it can go back into the nucleus and take NF-kappa B off the DNA. And this is another like, what kind of discovery, right? So this is NF-kappa B dissociating from DNA. Notice the time scale here. And this is NF-kappa B dissociating from the DNA when I-kappa B alpha is put with it. Okay, it strips it off. And we measured all the rate constants for this. But this is one of these things where, you know, oh my God, I mean, you need to get a mutant. I don't know if any of you have suffered through this. But protein, all the amino acids in proteins are doing more than one thing. But the reviewers want a mutant that changes just that one thing that you're saying that this protein does, right? And not anything else. And it was really, really hard for us to find that mutant, but we finally did. We found, one of my students, Holly Lindsay, she recognized that there was this whole set of negative charges at the end of the C terminus of the I-kappa B alpha. And so she postulated, hypothesized, I guess is the word we want to use, that, that these were kind of repelling the DNA. And so maybe that was what was causing the NF-kappa B to dissociate from the DNA. So she neutralized all those amino acids. She called this the 5X pest mutant. Holly did her entire thesis in pink and purple. So that's why these colors are how they are here. Okay, and so she did show that this 5X pest mutant cannot strip as well as the wild type. And the tricky part was we had to find a mutant that didn't bind any worse to NF-kappa B, because that folding on binding was a big part of the binding affinity of I-kappa B alpha for NF-kappa B. But here we are, we found this mutant. How am I doing? Okay. So anyway, we put that mutant back into cells behind the native promoter, and we showed that the export out of the nucleus is impaired. when you have that mutation in there. So what this says is that you know, orange green that I was showing you. The reason the nucleus, okay, the NF-kappa B goes into the nucleus and then it all comes back out. The reason it all comes back out is because of this stripping. It controls the rate at which the NF-kappa B gets taken back out of the nucleus. And then Peter did a whole bunch of theory to show this totally makes sense, because you have a bucket load of DNA in the nucleus. And NF-kappa B binds to DNA. So if you were just going along and making I-kappa B alpha and expecting it to go in there and compete with the DNA to get the I-kappa B alpha or to get the NF-kappa B out, it would never get it all out. And you would always still have cell proliferation going on, right? So you have to have these very exquisitely controlled systems where proteins do really cool things to each other to turn that thing off as fast as you turn it on in order to avoid bad things from happening. Yeah, so molecular stripping controls the rate of NF-kappa B export from the nucleus. And this is Peter's simulations, but I'll just skip those. Okay. Now, the last thing I'll tell you, and I don't think I'm going to get to the E3 ligase part, is that then we started doing single molecule FRET on the NF-kappa B. And like I told you before, the NF-kappa B in all the crystal structures is shown hugging its DNA in its belly here. But my student, Wei Chen, she took the DNA out and did some molecular dynamic simulations. And these two DNA hugging domains went like this, right? And so then we were like, oh my God, you know, maybe it really is hugging and unhugging and hugging and unhugging and, you know, right? So, so she did a bunch of single molecule FRET, and she showed that actually these two domains are at all different places with respect to each other. The distribution of FRET is super broad. You can have it all the way from total hug, all the way to way out here, and everything in between. This is not a two-state system. It's not either here or here. It's here, 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 right? Okay. And you know, we have all the traces. Here's one where it's sort of pretty far apart. Here's one where it's kind of in the middle. Here's one where they're close together. And then again, we saw these transitions. Okay. Here it is kind of middling apart. And then all of a sudden it goes further apart. Here's one where it's close together. And it goes all the way to far apart. Right? Okay, but blow your mind away. Then we did the experiment where we immobilized DNA on the surface of the microscope slide. And we expected to see that all that stuff would stop, right? Because the crystal structures show that when it's bound to DNA, it's hugging. No, look, we see the same kinds of traces. And mind you, the DNA is bound here to the microscope surface. So the only NF-kappa Bs that we are looking at in this experiment are NF-kappa Bs that are bound to DNA. And they are still going this way, right? And they're staying bound when one arm isn't hugging. And so we decided that this totally makes sense, right? Because otherwise, how would the I-kappa B alpha be able to strip? And the I-kappa B alpha, if you bind that to the NF-kappa B, what you see is the I-kappa B alpha forces the NF-kappa B not to be able to hug ever again. All the traces had very low FRET. All right, I think my time is almost up, isn't it? Should I quit? Yeah, okay. Let me just show you who did the work, and I'll skip the E3 ligase part. And another story for another day. There's a lot of interesting allostery going on in U3 ligases. I'll just tell you that. Okay, so this is my group during the pandemic. This was ways we have to have a party for everybody's thesis defense, right? So this is our socially distanced, masked, Chinese takeout out in Rebel Lawn party for Wei when she defended her thesis. Anyway, all right. You guys want to ask me some questions? It'd be great. Andrew.
Well, first of all, I can't wait to tell my therapist that frustration is a necessary condition.
Yes.
But also, I know when you first started your course, we first designed the course on molecular complex determination. It was very much in collaboration with the scientists.
Yes.
So like beneath that now.
Yep.
And I don't know if it's still the case.
So we always have Benit and or Nuno come and give a lecture because that whole thing is still, well, and okay, I will tell you some exciting news. Actually, hold on. Let me, yeah. Okay, Zoom is still here. What am I doing? End slideshow. Okay, I have to do. Not the one I wanted. Where's the lab? We just recruited Lisa Jones. Where is she? Well, let me go to her Wikipedia page. No, come on. Anyway, let me try Jones. She has a really cool. Okay, this is really bothering me. She has a Twitter. Anyway, I wanted to show you her lab. Oh, okay. Anyway, whatever. Lisa Jones. We just recruited her. So basically, let me just say that when we started that class, there was still a lot of work to be done to try to figure out what the best computer algorithms were to deep analyze proteomics data. And there's still some work to be done on that, but Nuno really got tired of it at some point. We worked a lot with him when we were doing sumo modification analysis because there's a lot of intricacies about how their algorithms work and how they score things and stuff like that. And when we were doing sumo analysis, we didn't have tryptic lysines and arginines at the end of our peptides, and so all the scoring went off, and we had to fix all that. But so now all of that is you know, there's there's several software packages you can buy that just do that. This is what happens with interfaces, right? You do interfaces and things get to be commercial, right? Okay. So Lisa Jones, I can't believe we managed to recruit this girl. She's really, really amazing. I'll just tell you, we got her because, I don't know if you guys heard about Isabel, no, what is the name of this girl? Have you guys heard of the 1619 project? Okay, so remember that there was this journalist from the New York Times that was recruited to UNC Chapel Hill. And then at the end, UNC Chapel Hill decided not to give her tenure right away. And so now she decided not to go to UNC Chapel Hill, right? Well, right during that time, Lisa Jones was also being recruited to UNC Chapel Hill to the chemistry department. And she watched what was happening and she was like, I'm not going there. And so then we have these faculty positions at UCSD for big famous African-American scientists to offer classes in the African-American studies minor and be in the home STEM department. And Lisa was like, oh my God, I would love to do that. So she's coming in July. And what she does is really awesome. In fact, Andrew, she's going to take over this class eventually because she does in vivo, including in tissues, she's developing this right now, fast protein oxidative footprinting. And I could give a whole lecture on this, but anyway, I talk about it in the 283 class. And so she can actually, she diffuses hydrogen peroxide into tissues, zaps it with a laser, makes hydroxyl radicals, and can label the surfaces of proteins on a whole proteome wide scale. And she's making all kinds of really neat discoveries about this. But anyway, part of the reason that she's coming here is because Nuno is going to work with her because her peptides are complicated, right? I mean, this is the thing. It's a really simple, if you understand this stuff, it's really simple. Okay, if you ask the probability that's modified is not very high. And so this is a huge problem, and Nuno wants to help her solve it. And so that's one of the main reasons why she came here. I don't know if that.
It's really exciting.
Oh my God, yeah. She's definitely going to be one of your interfaces people. Yeah.
But what I was also thinking is that sort of a similar thing has happened in we did molecular electrostatics, and then we went from there to body color model stuff. So now we do some molecular dynamics and some molecular electrostatics, and then Brownian dynamics, and then some Montana dynamics, and then some Monte Carlo modeling. And it occurs to me that we ought to be thinking about how we can sort of line up the different experiments of the different models so that students can be not only learning the data analysis and the analytical techniques, but also conceptually driven modeling as well.
Yeah. Yeah. And what you guys also need to realize is that there are so many gaps. There's like timescale gaps. There's, obviously understanding gaps, but and what I loved about what we discovered is you never would have hypothesized this, right? We never would have hypothesized that I kappa B alpha was ripping the F kappa B off the DNA. It was just a discovery that, you had to step back and go, oh, wow, it's doing that. So, there's still, so much of biology is like, we aren't even smart enough to make a hypothesis for it yet, So, I remember, who was it? I was listening to a seminar from some guy over at Scripps, and he's like, oh, I don't get NIH money anymore because they expect you to have a hypothesis. And there's a place in science for hypothesis-driven research. I understand why the NIH wants you to have a hypothesis, because otherwise they might waste their money on you, right? But on the other hand, a lot of science is discovery. And so, that's the other thing that I think is really good about the interfaces program is you learn how to play the game of, okay, here I can make a hypothesis, and then I use some of that money to fund my discoveries, right? And you can go back and forth. Anyway, I should shut up because the next person's up, right?
Yeah. Thank you so much.
you. Thank you. This is so colorful.
And Dr. Smar, thank you so much for joining us. Hopefully you're able to hear us with our owl and everything.
Thanks for having me. I'm really sorry not to be able to be there in person, but I'm very happy to join.
Yeah, absolutely. So hopefully you have the ability to screen share. And yeah, thank you so much for joining us. This is Dr. Smar, the new faculty member in the bioengineering department, as well as the Data Science Institute. For the last part of it, but I'm sure you will mention that as well. So thank you so much.
Okay. It looks like you guys, I can just see in your screen that you're seeing my screen, so that's good. So yeah, once again, thanks very much for having me. As he mentioned, I'm joint appointment between bioengineering and data science. And as you'll see, the way that marriage works is I do a lot of time series bioengineering. So it's data intensive and it's looking at patterns in time as opposed to, for example, patterns in structure like we were hearing about. I'm going to try to just talk through a couple of examples of how this helps us understand both the opportunities in the future, how the field is changing to enable precision health through things like machine learning, and also
Dr. Zainab Jahed, who's a new faculty member in the Department of Nanoengineering and an affiliate in bioengineering. And we're very happy that they're both here to share some insights. And we ask that you guys hold the questions until the end of each person's individual lightning talk. But then, of course, we invite the judges to ask questions as well as anyone else who has a question. And so with that being said, we will let Emma get started and then we'll pass out a couple of rubrics. Yes, of course. Yeah, it's right there. That's you. Yeah. Start recording.
Yeah, I think it is.
Oh, okay. Thanks. Chaotic screen share. It's just this one. Sorry, click start slideshow. Yeah. Okay. Let's do that. Yeah, that's right. You should be in business, and I think Stephanie, you timing?
Yeah.
All right. Yeah, feel free to
So as soon as after you introduce yourself,
Perfect.
Hi, everybody. Hopefully you can hear me okay. My name is Emma Boyd. I'm a third year in the neurosciences program at UCSD. And this is my second year in the interfaces program. I really enjoyed here. So I am very excited to talk to you about my research. Though I'm in neurosciences, my true passions are in biomedical imaging and specifically neuroimaging technologies like CT and MRI, all these like whole body or whole brain non-invasive techniques.
I know what that is.
On presenter mode. Okay. Wait, what? It just worked for you? Okay. All right. Well, okay. So for this talk, I kind of wanted to just boil down my research into one core question and give you some background for what I'm studying. So my research centers on studying hemodynamic change in aging and Alzheimer's disease. And if I want to, think about one big overarching question, it's basically how does white matter perfusion change in aging and Alzheimer's? The motivation for this work is that obviously, I'm sure as everybody probably knows here, Alzheimer's disease is a really terrible neurodegenerative disease marked by a really progressive cognitive decline and dementia. There is an extremely large population of aging individuals who are going to be getting the disease, especially as boomer generation ages. And so researchers are really racing to identify a biomarker that's indicative of early Alzheimer's disease process. And so they're looking to find something that you could identify maybe middle-aged individuals before progressive anatomical change and neuronal loss and eventual cognitive decline occurs. And in my area of research, researchers think that perfusion changes in the brain, which I'll describe that in a second, but perfusion changes could be a really early marker that is indicative of that eventual neuronal loss. And perfusion is the process, I have a little thing there. Perfusion is the process by which nutrients are delivered to the tissue bed. This is a process that occurs in any body tissue, but when we're talking about the brain, this is especially important because it's useful and completely fundamental to neuronal activity. So when the brain is active, perfusion tends to increase so that more blood and thereby more nutrients are brought to the brain so that it can be metabolized and waste products can be removed. It's fundamental because what happens when perfusion declines is that tissue dies, very simply. Like you can't survive without adequate perfusion and adequate nutrients. And then what we find in age is that generally there's a progressive decline with age. There's regional reduction and regional increases of different parts of the brain. Is that standard in vivo imaging techniques have certain biological and methodological limitations that prevent the accurate quantification of white matter perfusion. WM is white matter here, GM is gray matter. And the two biological limitations are that number one, white matter is located at the distal ends of the cerebral vasculature. So when blood is flowing upwards from the carotids and the vertebrals, it kind of flows in a couple different loops that first perfuse the gray matter and then go deeper and perfuse white matter. So it just takes longer for blood to get there. Furthermore, white matter also has a lower vascular density, so just less blood flows there, and it takes a little bit longer to get there. This concept of transit is very, very important when you're talking about in vivo neuroimaging techniques, because this transit is longer for white matter, so you have to kind of alter your techniques to accurately capture perfusion. And furthermore, we also think that white matter is more vulnerable because of this transit issue, and therefore it could be an even more specific biomarker when you're talking about Alzheimer's and aging. Okay, so the technique that I use is called arterial spin labeling MRI. It's a technique by which, it's a whole class of techniques by which arterial blood is magnetically labeled. That's why the label is in quotes there. In standard techniques tends to be labeled at the base of the brain and that giant red line there or in a big slab. So there's a volume of blood that's labeled. There's a transit time by which the blood flows up into the brain and reaches the capillary bed where it perfuses tissue. And then at the blue area there, you acquire signal, you take your images over a region of interest. And the signal that you get is the labeled blood water that accumulates within the tissue from crossing from the blood into the cells and the space in between the cells. What you do with this type of data is you apply a kinetic model, which very simply is just an equation that takes the signal magnitude that you get from your image and relates it to a measure of perfusion so that you can tell how much blood has actually reached the bed. I'm running out of time here. But the whole crux of my work is basically taking these standard techniques that in aging and in diseases, surprise, surprise, do not work because of vascular changes in aging. So if you have something like atherosclerosis, or if you have a tortuous vessel that takes longer for blood to get there, then you get a lot of different regions of hypoperfusion or regions of hyperperfusion that are actually artifactual. So in my research, we use a new tool called velocity selective ASL that labels blood actually within the imaging volume itself, thereby negating the need to have this label enjoyed. So you actually get what looks like real data. And then I'm going to skip over, but basically, in short, what we're doing is developing ESASL to be better, to be more accurate for white matter perfusion, and then we're going to apply it to several different aging populations. Thank you, and apologies for going so over time there.
Any questions? Yeah, go for it.
We also say them all towards the end or something.
Sweet shirt.
Hi everyone, my name is Kristen Garcia. I'm a second year PhD student in the bioengineering department, and this is my first year in the interfaces program. And today I'm going to be talking about my current research interest, which is sex-dependent effects of on right ventricular remodeling during pulmonary arterial hypertension, but more specifically estrogen's effect on this remodeling process. So first, we're going to talk about what pulmonary arterial hypertension is. It's a life-threatening progressive disease that is characterized by pulmonary vascular remodeling and is represented by the narrowing of the blood vessels in the lungs. So the pulmonary arteries are the blood vessels that carry blood from the right side of the heart throughout the lungs. And then a little bit about the prevalence of the disease. So in the United States, there are about 500 to 1,000 new cases of PAH each year. And this disease is most common in women between the ages of 30 and 60 years old. And in fact, women are 4 times more likely than males to get this disease, but however, they're also way more likely to survive the disease than any man that gets it. So this leads to an interesting paradox around estrogen that is being investigated today. Past research has shown contradicting results though. Some have shown that estrogen has a protective effect over right heart failure, but others are showing that estrogen is the disease promoting effect and is causing the disease. So more research is needed around what estrogen is actually doing to right ventricular remodeling. So my goal is to determine the role of estrogen in PAH, but more specifically the role of estrogen on the right ventricle. So it's been found that the right ventricle initially adapts to the disease by increasing muscle contractility and wall thickness to compensate for like the pressure overload. However, this reaches a point in which it goes into severe decompensated right heart failure, which leads to a reduced cardiac output and wall stiffening. And the point in which this, the right ventricle goes from this adaptive remodeling state to this irreversible maladaptive remodeling state is unknown. So the whole process through which the RV remodels has yet to be fully understood. And I'm specifically hypothesizing that estrogen plays a major role in the remodeling of the right ventricle, but more specifically, it has a protective effect against right heart failure. And I aim to do this by quantifying the hemodynamics of the right ventricle through an animal model of PAH. And then my end goal is to create a biventricular mathematical model to help understand questions around the sex differences in the remodeling process. So a little bit about my method. So first, we're using a Sugen hypoxia protocol to induce pulmonary arterial hypertension into the rats. We use both male and female rats, and there's 2 groups of female rats, one group with their females intact or their ovaries intact, and then one group with their ovaries removed to represent pre versus post menopausal women. And then once the disease is induced, we perform terminal surgeries on the rats. And so you can see in box B, this is a representation of the surgery, and these are pressure volume catheters that are going into the right ventricle of the heart to produce these PV loops right here. It's kind of hard to see, but this is a PV loop. And from these PV loops, we can get different metrics to see how the heart is functioning. So we can see heart rate and systolic pressure and volume and diastolic pressure and volume and can use these metrics to calculate stroke volume, ejection fraction, and cardiac output. So all can tell us how the heart is functioning. And then after we get all these PV loops, we harvest the heart and run it through a planar biaxial testing protocol. And so this can get us information about stress strain measurements and can tell us how different tissues have different stiffnesses and how that's relating to the progression of the disease. And then lastly, we run it through a histological analysis to examine any alterations that could be made on a cellular level, for example, like different collagen contents or ECM matrix in general. So I'm currently planning to dive more into the estrogen component of this remodeling process. So it has been found that the rats go through an estrous cycle that is similar to a menstrual cycle in humans. However, you can see the lengths are different. So each stage, the lengths are shorter than in a human. But pretty much what we have found is that throughout the progression of the estrous cycle, there's different levels of hormones in the rats. And so this is crucial for our studies because if we're performing a surgery on a rat at a specific stage and we don't know what the stage is, then our conclusions can be off based on the stage of the rat. So my overall goal right now is to determine a way to measure the estrous cycle stages in the rats. And so we can relate then the estrous stage, the level of estrogen, and then the RV function as a whole. And so with all this in mind, in conclusion, so I'm pretty much trying to help solve the estrogen puzzle and see what is actually estrogen's main role in right ventricular modeling. And I hope with my mathematical model, I'm able to help understand why the function of the right ventricle for males and females is so different for the same disease. And that's it. Yes.
So, do you know how much of the difference in between males and females in the right ventricular remodeling is due to differences in the interface, in other words, due to the vascular?
Yeah, I'm not really sure because we were trying to figure out if it's due to just the vasculature in general or something specific within the hormones.
So, it might be interesting for you to look at your data and maybe sort of do some sort of correction or account for the difference in nutrition to see if the differences in remodeling that say are still there if you don't if it's correct for adjust for the difference.
Yeah, that's super interesting. I'll look into that. Thank you.
So morning, everyone. I'm Marcus Oak. I'm a PhD student in the bioengineering department. This is my second year in interfaces, although it sometimes feels like my first since things are finally getting back in person. And I've been using multi-scale simulations to understand the effects of DATP on actin-myosin interactions, as well as some myosin mutations on the interactions. So very briefly, what is a DATP, or 2'-deoxy ATP? It's a near analog of ATP, and you can see that it's almost identical with the exception of just the hydroxyl group is removed on the DATP molecule from the ATP. And it's also an energetic substrate. And myocytes with increased concentrations of DATP generate greater amounts of force and actually have increased shortening. And so as a result, DATP is proposed as a heart failure therapeutic to help restore the function of the heart. So dilated cardiomyopathy is one of many causes of heart failure. Dilated cardiomyopathy has also been linked with the myosin heavy chain in terms of different mutations. And so DCM is characterized by weakened ventricular function, there's reduced wall mass, dilated ventricular chambers, and overall progression to an ultimate kind of heart failure state. And there's two particular mutations on this myosin heavy chain that I'm most interested in. There's the alanine 222 threonine mutation, as well as the serine 531 to proline mutation. So you can see I've plotted those. You actually can't see it there, but the alanine 222 mutation is this yellow one adjacent to the nucleotide binding site, and the serine 531 mutation is kind of on this actin binding surface of the myosin cleft. And so the goal is to use computational models to scale kind of across size scales, and understand the mechanisms of these mutations, as well as DAPP and how they lead to altered function. So these simulations kind of begin at the small scale using molecular dynamics simulations. And so we're able to simulate myosin in this pre-power stroke state at the start of the crossbreed cycle. And from this, we've been able to construct a reduced Markov model of 15 different conformational states of this protein based off of the motion of this actin binding surface. And so using these models, we can then actually take each of these different conformations and run those in running dynamic simulations to understand interactions between actin and myosin, which is crucial for the function of the crossbridge cycle. And so it's this kind of first step in the crossbridge cycle that starts muscle motion and the mechanical lever arm that you might be familiar with. So I want to first start off with just kind of analyzing this Markov state model and the conformational dynamics between these different simulations. And so one way to visualize a Markov model is using a transition matrix. And so basically what this demonstrates is a heat map that shows the transition from any particular state to an adjacent conformational state. And so each one of these, you'll see along the diagonal, you have kind of the brightest heat map, and that's because you kind of increase the probability of remaining within the same state as opposed to transitioning to a different state. But what I want to highlight is that DADP actually has kind of this brighter diagonal compared to all the other conditions. And so this kind of suggests the fact that DADP establishes a more kind of stable conformation. It's less likely to transition between slightly different conformational motion, kind of like Dr. Cumens was talking about this morning. And so when we then take these conformational models and kind of run them with a Monte Carlo sampling and Brown die, we're able to generate what we call a reaction curve. And so within Brownian dynamic simulations, you have kind of this parameter that's adjustable, and this is your reaction distance. So this is basically defining how close these two molecules need to get before they're defined to have reacted. And so we run this across a wide range because we don't know exactly what distance is correct, but what we can look at is experimental measurements. And we can say, what is the wild type association rate of myosin and actin? And if we find that this is about 2.5 times 10 to the sixth inverse molar seconds, we can then say that that's a reaction distance of about 6.9 angstroms, and then we can compare accordingly at that rate distance. And so ultimately what we see is that DADP has about a 2.7-fold increase compared to ADP in this association rate, as well as 531P is almost threefold increased compared to ADP, whereas the A22T mutation is actually decreased by about a factor of a half. And so this is really interesting because these are both DCM mutations, but they lead to completely different phenotypes. The last thing that I want to show with these Brownian dynamic simulations is kind of the change in conformational states. And so you'll notice that each one of these curves now represents a different conformational state. And so within these states, there's actually a really wide variance in terms of the association rate. So this just goes to show that there's one state that might lead to one curve. There's up to like a fourfold change between these different conformational states. And so this is something that's really important to kind of account for when we're modeling these association rates. And so we want to account for the full kind of dynamics of the protein. So lastly, kind of some key conclusions. Both the DADP as well as the S531P mutation lead to an increased actin binding association rate with myosin, which is opposite from the A22T mutation. And this kind of demonstrates the fact that maybe DADP can reverse the effects of this A222 mutation and restore normal function. And lastly, I kind of want to reiterate that there's these subtle changes within the molecular dynamics simulations that lead to significant differences, as well as the fact that DADP appears to stabilize the SM head. So almost 5 minutes and some acknowledgements, and I'll take questions. Thank you so much. Yeah.
Sorry, I missed it. Could you just re-explain real quick those two mutations? Like, why are they interesting?
Yeah, absolutely. That's something that I would have wanted to mention, but 5 minutes is short. So the reason that we looked at kind of this A22 mutation is the fact that it's adjacent to this nucleotide binding site within the myosin motor domain. And so you can see kind of here's our nucleotide here. And so the A22 mutation is right here. And so we might see some interactions between DADP compared to ADP that's differing there. So kind of intrigued by that. And then this S531P mutation is kind of on the surface of the protein that interacts with actin. And so we're curious to see how changes there, since that's going to be the part that directly binds changes functions. And again, both of these cause dilated cardiomyopathy. So they're both potential, causes of heart failure, and we might want to treat them.
share screen again. Okay, I'm Nate. I'm a second-year PhD student in the mechanical engineering department, although it feels like my first year since I moved here this fall. Today, I'm going to talk about my work on Bayesian parameter estimation from sparse and noisy measurement data in systems biology. So my work presents a framework for parameter estimation in systems biology. There's kind of three main steps. So there's this model development step and data collection. There's this pre-processing and actual parameter estimation step, and then there's this simulation and output uncertainty analysis. And Bayesian parameter estimation really enables this third output uncertainty analysis step. So if you can think about experiments, you do multiple trials, and you can measure some statistical significance of your results. In modeling, you usually get one trajectory of results, and you can't do that kind of statistical analysis. Bayesian estimation and uncertainty quantification give you a framework to do that sort of quantitative analysis. So systems biology has leveraged mathematical models to quantify biochemical systems, and in this case, the dynamics of biochemical systems. So we have these differential equation models that have some equations and some parameters theta. And my goal is to learn these parameters from measurements of the states. So some forms of these equations are the typical mass action kinetics, Michaelis-Menten kinetics, or Hill equations. And a quick example of what these models looks like is this simple MapK signaling model, where we have 3 states and 3 differential equations, but 14 parameters. And this mismatch between the number of parameters, the number of states, and likely the number of measurements, maybe we can only measure state X3, makes parameter estimation very challenging. So given that we have a lot of parameters, not a lot of data, we need to ask what parameters can we actually estimate, and of those parameters that we can estimate, are they actually meaningful for the model? And by meaningful, I mean, if we change it, does it actually change the predictions the model makes, or does it have no effect? And we can use these 2 analyses called global identifiability analysis and global sensitivity analysis to answer these 2 questions. After these 2 whole, I could give 2 whole talks on these different analyses, we get a reduced set of parameters that we can actually estimate and that are meaningful to estimate. So then we want to do the actual estimation. And rather than assuming each theta can take a single value, we assume it can take a distribution. This distribution is called the posterior distribution, and it's the distribution of theta conditioned on the data we measure. And we use Bayes' rule to express this distribution from the prior, so that's what we think the parameters can be, and the likelihood, which measures the mismatch between model simulations and the data we collect. And this UKF MCMC algorithm is a way to estimate the likelihood with an unscented Kalman filter, that's the UKF, and to draw samples from the posterior using Markov chain Monte Carlo sampling, that's the MCMC. So when it's all said and done, I get a collection of samples from the posterior that represent the distributions of the parameters. I can use those samples to enable this output uncertainty quantification. So the simulation step is I take the samples from my posterior and I simulate the system, and I get a collection of trajectories. Then I can do things like quantify uncertainty intervals. I could look at a distribution of the steady state values that we get and use that to help with the biological.
Or kind of what do you do once you learn what's most important?
Yeah, so I do identifiability analysis. The ones that are not identifiable, which is a mathematical property of the model, I fix to, I call it the nominal value. So maybe it's the middle of the range that I assume it could be from previous literature or what I think it could be. Then the ones that don't produce significant variations, the output I also fix. And then the other, that subset of identifiable and sensitive parameters are free, and those are the ones I estimate. Cool.
Okay, hello, everybody. Oh, do I have to put it on?
Yeah.
Oh, I see. So no one sees it. I see, so it comes out every time.
Put it in back and present. Go to, or just go the way you present that.
The way I just had it, yeah. Okay, thanks. Cool. All right. Hi, everybody. My name's Sean, and I'm in the Machinina lab, and I'm in the biochem department. It's my second year in the interfaces program and my third overall. And I study phosphorylation and acetylation of this protein called mitochondrial transcription factor A. And in my talk, you'll see it called TFAM, just to keep it short. And how it exerts effects over transcription and compaction, basically by modifying it, these modifications. Okay. So you all know you have mitochondria, right? Not everybody knows that your mitochondria has its own DNA. And this DNA has to be transcribed and translated into really important proteins, especially for oxphos, which produces your energy. And I study this protein, TFAM, that actually binds and compacts DNA. So it protects mitochondrial DNA. It also makes it very small, so it fits in mitochondria. And it binds and bends it. And so, yeah, we want to know, how does it basically decompact and compact back up, right? So you have to do that to control transcription and replication on mitochondrial DNA. So it also has one other really odd function. It's a transcription factor, just like its name, and it recruits the polymerase for transcription. So how does it have the two completely opposite roles, if you think about it, being a compaction protein, regulating transcription, but also helping transcription initiate in the first place? So there's two possible modes of regulation. One is obviously its expression, right? It's a nuclear encoded protein, so its expression will change what the nucleoids look like, right? But also my big hope is to look at post-translational modifications, and that maybe being a more local, also really quick way of changing nucleoid, which is what they're called, mitochondrial DNA are normally called nucleoids, structure. So the two big post-translational modifications that have been found in vivo so far are acetylation by acetyl-CoA, non-enzymatically, and phosphorylation by protein kinase A, as well as other kinases, but I studied protein kinase A just to start. So I had to make a stock of TFAM. So I purified TFAM from E. coli, and I purified protein kinase A as well, and you throw them together with ATP, and you will have phosphorylated TFAM. I use mass spectrometry to find the sites, and just a really quick estimation from Max Quant of how much one site is particularly phosphorylated versus unphosphorylated in my stock. You can use this intensity mod over base, which is a sum of the modified peptides, the intensity of the modified peptides in your sample, divided by the sum of the peptides for a specific site that's unmodified. So I also reacted TPM with acetyl-CoA to acetylate it, and used kind of a little bit of a different, more quantitative way of finding out how acetylated a given lysine is on TPM. There's many lysines that could be acetylated. So in this normal workflow of mass spec preparation, you have a denatured protein and you react it with a very strong acetylating agent, which in this case is heavy acetic anhydride. So the acetyl group will be heavy labeled. And you can see basically the light group would come from acetyl-CoA in my reaction, and therefore in my stock, that lysine is acetylated versus, what I get back in the mass spec will show me, the proportion of that lysine that's not acetylated. And so you kind of get this sort of, this sort of distribution here. You get kind of like a light and then a heavy right next to it. This is all one, this would be one given peptide. So you have to quantify for each peptide for each site. And you have to add this as a custom modification into Max1, which is really nice. So, yeah, and in short, I saw these residues are heavily acetylated, 52, 154. I only found one significant phosphosite, which is pretty interesting. So pKa only recognizes that spot as, a motif for phosphorylation. And so I tried to use these stocks. So I tested, can it compact DNA, right? Once I have these stocks, they're kind of characterized. So you compact just PUC19 plasmid DNA and you run it in a gel called an EMSA, and you compare the shift. So yes, it appears that, the control versus acetyl versus phospho-TPM, each one compacts, which, a hypothesis out there is that it can't bind DNA when these things have happened. And that's not true, which is pretty interesting. I thought this was a pretty bad result at first. I then tested whether it can initiate transcription. So can it still pull the polymerase to the right spot for transcription? The answer there was also yes, which is pretty cool. I then tested whether or not the polymerase, PolRMT, can plow through DNA that's covered with TPM. So this would be more of the compaction regulation aspect by making this tail that PolRMT can start without TPM. So it's kind of removing that need for TPM as an initiation factor. And then you measure the RNA output you get. So if you have a high level of compaction, TPM knocks down transcription. When you have acetyl TPM, that transcription is upregulated. So therefore, it's more easily removed by the polymerase. So even though it can bind DNA, it has subtle changes. And it's even more pronounced with phospho-TPM. So I'm presenting a new model for how it's regulating, and it can still compact, do that function, but it changes other functions of TPM. Thank you.
Okay. Hello, everyone. My name is Joseph. I'm in the Department of Chemistry and Biochemistry. And I do quite a bit of chemistry, actually. So I think it's really cool how this program brings all of us together, people doing like mathematics and bioengineering and stuff like that. My research advisor is Professor Jerry Yang. And the research I'm interested in is the design and synthesis of novel agonists for the treatment of gut disorders. Agonists and activators I'll be using interchangeably because they are synonymous. So my work involves inflammatory bowel disease, IBD. And this is an umbrella term of gut disorders. Most commonly known is Crohn's disease and ulcerative colitis, or ulcers in your colons. This is affecting a lot of people, 1.6 million Americans. The average age of diagnosis is very young, 21 years of age. And on average, 2 surgeries, usually very painful, like taking out the entire colon. So obviously this affects a lot of people and significantly. So I wanted to go over how the collaborators that we've been working with, Professor Pradipta Ghosh in the School of Medicine, has been working on identifying a target for IBD. So what they started with was a large data set of mRNA transcriptome of many human patients that have Crohn's disease. And through this data set, they started to begin to, they joined together clusters of genes that are very similar. They used relationships between these different genes to create the clusters. So for example, someone with Crohn's disease might have a gene A expressed low and gene B high, and so on. And these relationships were then mapped out on a continuum state throughout the normal to disease state. So for example, cluster 1 might have a very high gene expression level in someone who's very healthy, and very low, but cluster 5 might have a very high gene expression level in someone who's very diseased. Another way of looking at this is seeing the different gene clusters and where they're at. And I know this slide is a lot going on, but basically it's showing that our collaborators were able to find a target protein, PRKAB1, in gene cluster 2. This is one potential protein target among many of the protein targets that they're able to find. PRKAB1 is the gene that encodes for AMPK beta 1. And so specifically, that is where my research starts, and it's the crux of my research, how to target this protein. So my work involves the design, synthesis, and testing of novel beta 1 activators. And so to start with the design method, we used in silico docking and X-ray crystallography. So to credit my colleague in my lab, Ashish Shivkumar, he was able to look at many of the different proteins that are already published of X-ray crystal structures. So here I show a crystal structure of this molecule here. It's a known beta-1 selective agonist made by the Pfizer company. So that's why it's PF. And it sits here in the pocket of AMPK. Beta-1 is one of the isoforms of AMPK. So it sits here in between the beta-1 and the alpha-1 subunit, the allosteric binding site. I have here highlighted the black protein residues, which are interacting with that drug. Red residues are ones that are surrounding the pocket and can be potentially used to interact with the drug, creating higher selectivity and higher binding. So a question that I have is what makes this molecule a beta-1 selective agonist over beta-2, as well as other beta-1 selective molecules? So using more than 100 known molecules with crystal structures, we are able to look at not only novel agonists, but known beta-1 agonists. I don't have that shown here, but the design that we used was to interact with additional protein residues here in the pocket while keeping the pocket shape the same. So this molecule here, which I just note as PF derivative, you can see here that when it's docked into the binding site, it keeps the same shape of the binding site, and it keeps many of the similar protein residues interactions, but it also reaches out with this ether side chain and a carboxylic acid with arginine 83. So that's very important, and we predict that that will increase the binding selectivity and the activity when bound. So my favorite part is the chemistry. So I'll just go over this real quick. This is the known published chemistry of the known molecule. And I guess I'm running out of time here, but you start with the 1,4-dibromo-benzene and add the cyclobutanol right here, and then through Suzuki coupling, create the hydroboronic acid. And then basically it's to use these two pieces, molecule 2 and 3, and combine them with the indole scaffold to make compound 4, which is then furnished with the hydrolysis reaction. So for me, I had this basically synthetic scheme mapped out, and I had a lot of troubles with getting compound 5 to turn into compound 6. I was using an oxindole to nucleophilic attack this molecule here, but I had a lot of issues with that. So using a separate method as the this being the nucleophile and attacking with this 3-chloro intermediate, I was able to form this product, which I'm very excited about, and will be able to lead me to molecule 8 very soon. So yeah, and then after that, I have plans on testing in vitro using this assay, and I don't think I have time to go over that, and that's about it. Thank you, guys. Yeah, does anyone have any questions? All right, thank you so much. You have an announcement, Marcus?
It's lunchtime. Well, it will be soon. So I think officially it was 12:45, which we, lightning talks quickly. So I think we can mingle for a bit. Feel free to stretch your legs since we've all been sitting. And I think the food is either being brought up now or will be in the near future.
Actually, before we have lunch, I'd like to get a picture outside. I also want to thank all the license for excellent. It's actually hard to get by, but I thought very much to hear about I do have one little request for lunch, and that I say this all the time at different places meetings. We tend to sort of say when we introduce ourselves and talk about the lab that we belong to, and we belong to, but in interfaces we'd really like it if you acknowledge that co-mentor. It's not that as a mentor I need my name, but just want the co-mentor part of the program to be emphasized. So it's really a key feature of this program, and sometimes these things get a little pro forma. Get it, because most of our time in life.
Yeah.
If we go outside the limit,
You said you primarily use microscopy.
Yeah, she like her PhD was in microscopy and then she does like confocal and all that stuff.
Nice. That's cool. Yeah, we at my work, it's all microscopy based, so it's like I have a big appreciation for how tricky it can be now. So it's like very, very cool to hear about.
I'm just curious, which microscopes do you use? Do you use the Zeiss most or do you use the?
It's not that. It's I don't remember the name. We only recently started like moving our microscopy in house from like because we've been using outside firms before because they're like so good. I am not the right person to talk to about our lab hardware.
No,
That's
I was
like
just
fairly
saying.
good.
My wife was like, oh, you do that? What she would be asking me what microscope.
No. Unfortunately, it was fun. Michael.
Kevin.
Yeah, nice to meet you.
Nice to meet you.
Hi, I'm Emma.
Kevin.
Sean and I are moderating, so waiting for you to come back.
Awesome.
Hey, Kevin. Nice to meet you.
Just gonna ask you guys to just give a short intro on yourself, if that's okay.
That's good.
Hi, everyone. All right, so we're going to get started with the industry development panel. Sean and I will be moderating, and we have our three panelists here. We have Kevin Vincent, a data scientist at Alphatech Spine. We have Michael Wiest, a data scientist at Spring Discovery, and we have Britton Boris, a senior principal scientist at Pfizer. So please give them a welcome round of applause, and I'll have them say a couple words about themselves and their path into industry, and then we can get started with some questions.
Right, Kevin Vincent. I did my, obviously I was part of the interfaces program at UCSD. I was in Integral Codes Lab, and I left UCSD about a year ago, and I've been working for a spine company, Alphatech Spine. I work in their neuromonitoring division, kind of their technology division, working on a neuromonitoring device. So I develop algorithms, including machine learning algorithms that help monitor the health of a patient's nerve during a surgery. So that's what I've done.
Cool. Yeah, my name is Michael. I got my master's at UCSD, I think maybe 4 years ago now. I was in Karsten Zangler's lab, primarily doing microbiome research. Now I am at Spring Discovery, where it's a drug discovery company using fluorescent microscopy. And primarily now I use like deep learning techniques to better represent human immune cells in like some compact numerical representation so that you can understand if drugs are doing what we want or not. Good.
I'm Britton Boris. I graduated out of Andrew McCulloch's lab. I was also an interfaces student. I now work at Pfizer. I've worked there for about 6 years. I do PK/PD modeling, so it's basically a lot of mechanistic modeling of predicting how our preclinical experiments will then translate to the clinic using computational models, mostly in oncology, but I've also done some work on COVID in the last 2 years or so as well.
Thanks very much for sharing. It's great to have some former interfaces people here and, to give some advice for our trainees.
Should have done it, sorry.
Should
have joined.
Yeah, well, maybe never too late. So I guess the first question that we kind of have here is, number one, I guess, why industry? Why did you move to industry? And number two, how did your graduate work at all kind of shape your trajectory going into industry? Do you pull anything from your past in your current work? Or is what you're doing now completely different than what you did in your graduate studies? Start with whoever wants to take it.
You got it.
Sure. The why industry, that was the first one.
Five.
Why industry, I mean, probably if you'd asked me 5 years ago, like which of the two panels I prefer to be on, it wasn't this one, you know. But just a lot of it for me was that, you know, having an academic lifestyle is challenging. So I have a young family, and you know, my first kid right about the time I was finishing my PhD, and my wife made a lot more money than I did as a graduate student, and so it was really hard for me to be like, hey, let's do a postdoc and move, you know. And I had an exciting opportunity just to stay here and continue working in Andrew's lab, and that was kind of the direction that I stayed for a while. And once you know you know, the academic path kind of closes off, you know, after a while, and it can be difficult to do, especially when you have, you know, a family to consider. So it's a balance, and that's I don't think we're not, you know, COVID, I don't think did a lot of great things for the academic job market either. So the writing was on the wall for me, and I really feel very fortunate where I've ended up. A lot of the things that I enjoy about academia, I still have in my current position. So I feel very fortunate.
Yeah, sort of similar answer. I mean, like in some ways, like it is like about money, right? But I think also, like Kevin was alluding to, like I don't really think there's that much of like a dichotomy in terms of the work that you do. Like I think, at least sounds like based on your guys' work, like we're probably very fortunate to do like research effectively at work, right? And I think, but there's also a lot of ways in which the companies have like more money. So it's like, if you need to spend, like you were saying earlier, it's like, oh, if you need to spend like $500,000 on a software license, right? Like that's not like a deal breaker, right? Like you can go to the company and they'll be like, sweet, sounds good. Like that's worth it in some situations, right? Like, but like, I think so like beyond like the personal money angle, I think like there's certain, like the company having money also enables you to do interesting research that might be prohibitive in sort of academia alone.
I mean, I personally, I always wanted to go into industry. I worked for 3 years between undergrad and grad. So I knew that I liked industry. I like the collaborative nature of industry. I like the like feeling like I'm really like right at affecting the clinic, seeing effects on patients' lives, feeling like my work is like immediately having an impact. That's a great feeling. And it's one I wanted. So that's why I went into industry, and that's where I want to go. To take one of the other pieces of your question, I don't, it's interesting because like I did my entire PhD was in heart modeling, and now I do almost entirely oncology. So in that way, they're not connected, but the tools that I learned, the computational methods that I learned, the thinking and the processing that I learned got me the job, and I use every single day. So in that sense, what I learned in grad school was essential to what I do right now, even if the biology is a little different.
Yeah, I totally agree. Like I think in terms of things you learn, I think it's like the soft skills are like super duper important. Like how do you communicate with people? How do you like ask the right questions? And like, at least where I work, like I'm on the computational team, and then we have a team of biologists, but it's like we're super enmeshed, and it's like being able to have the right vocabulary and like know and like be humble enough to be like, I'm gonna ask a stupid question, and it's like that's okay, right? And then like they don't care. And so I think like those sort of like paper reading and then like communication skills are like broadly transferable.
The follow-up, I would say, yeah, I did all cardiac work, you know, before I went to industry. Now I work on, you know, neurophysiology and neuromonitoring, which was a big pivot, but the problem solving, definitely, you know, the problem solving and the ability to collaborate, I think, which I think is the collaboration is an important part of the interfaces program. Like an important thing that I picked up as being part of that program. You know, I feel like that they're essential to my position, right? I collaborate with a lot of different groups. I'm probably a bit more towards the development end of things. I'm on the research team, but you know, my algorithms that I developed are in a device and in the operating room right now. And so like I have to work with the software teams that are going to develop, you know, implement stuff. I work with the systems teams. I work with the people in the field who are using the algorithm to get, you know, in the operating room to get feedback from them. Being able to collaborate with all those different groups and kind of speak somewhat different, you know, technical languages to those different groups has been important. And just problem solving skills, like just being able to have an open-ended problem given to you and be able to find your way to a solution of, in a methodical way that you can tell people what, the steps that you're going to try and take is, I think, something you pick up in grad school that you'd be kind of surprised sometimes. Like that's not something that's not the way everybody thinks. A lot of people want a ticket that says exactly what they're going to do for their work that day, right? And that's not really the way that I learned to solve problems.
Did we miss anything?
Okay. Totally covered it.
That's great. You guys all had great answers to that, and all different answers too, from knowing you want industry right away to kind of having to, find your way to it. And we have tons of other questions that are really interesting. I think we'll have different answers as well. So a lot of people that, we ask questions for are usually graduate students who don't really know what industry even means. I would say that about myself as well. I just don't really even know what it looks like, and I think the main fear of a grad student entering industry is that, this kind of if they wanted to learn an experiment or how to do a technique, they would, they're only good at one technique now and you don't get to expand, your toolbox anymore, which is kind of a question I have for you. Is that true? Is it restrictive in that way where you got hired for a specific skill and you aren't going to get to expand a toolbox anymore? And you're kind of more on a, you're kind of a cog in a machine essentially. I think this is a fear that I just want to address. I think it's a fear. I'm curious about how realistic that fear is.
Can you grow in your job?
Yeah.
Yeah, sure. I'll take it. I mean, I think maybe it depends on the type of company that you join to some degree. I'm at maybe say a medium-sized company, but I'm one of the only data scientists, right? And the data science is the growth area for the company. So I feel like there's plenty of room for me to grow. When I come up with kind of new initiatives and pitch them to people, I feel very supported in that. And I certainly don't feel like there's a direction that I couldn't grow in if I wanted to. But I understand that fear for sure. And I don't know, I'd be interested to hear from Brittany, who's worked, at a much larger company, if that's more of a challenge for you.
I would say that I'd frame it like this, or I'd say it like this, that when you go into industry, I was completely in your same boat. I was like, how do you what is a day like in as a grad student? I was like how is this a job? What does this mean? A lot of times it's a lot of collaborations. It's a lot of discussions and meetings, and then bringing your specialties to the table. I have never felt like I couldn't grow. It was always, but I would say you're growing in certain ways. So for example, and it's a lot about what job you choose to take. For me, my PhD was half experimental and half computational. I have not gone into a lab in the last 6 years. So in some ways, that means that experimental side of me is not there anymore in some ways, but I still use the knowledge base because I have to now tell other people, okay, we need this experiment to go like this, and these are the controls we have to run, and this is, that discussion really helps me to know what the experimental side needs. But then it allows me to grow in computational modeling in ways I've never thought about before, in areas that I had never even considered in my PhD. And I think, at least for Pfizer, and I think it's probably true for most other companies, is that if you're hiring someone coming out of school, you're going to teach them half the skills they need. You're not, the assumption is that you have the core, but you're going to need to learn a lot. And so expect to learn a lot in the first 6 months, how to talk to people, how to speak their language, how different techniques that you never needed in grad school. So personally, if I ever felt that stymied to quit, I'd go find a different job, and I haven't yet.
Yeah, I kind of agree with both of you. I work at a fairly small company, so it's a small team. So it's people are you wear a lot of hats, but it's super true. And so I'm constantly learning new things, and not only learning from papers and resources online, but so much from my teammates. And I think that's something to keep in mind when in industry is you have like really, really close collaboration, at least on a computational team with your teammates. And they all have so many skills that I'm like constantly picking up and growing in ways where it's like, I'm in awe of like the software engineering side of my team. Like they're such killer engineers and I'm like constantly trying to learn from them. And yeah, I think to your point, it's like, there's like some adage where it's like in your first week at like a programming job, you learn more than all 4 years of like your undergrad CS degree. And I think that is super true even in like kind of the biotech space as well. One thing is like, I would say is like, maybe you guys have experienced this, is like, if you want to keep growing and you want to like kind of change or divert what you're learning or your career, like you kind of need to advocate for yourself in industry a little bit. Like if you have a manager or, you know, someone who you report to, like, it's really important that you be like, hey, I'm not happy in this situation, or hey, like I want to learn this thing more. And at least in my experience, everyone's always really receptive to that. And it's because like, they want you to be happy. If you're happy, you stay. They don't have to hire new people and you keep like being productive, right? And so I think in every situation I've experienced, like if you speak up for yourself and say like, I want this thing or I need this change, as long as it's reasonable, it'll probably happen, I think.
And I think that and this is a cliche, and I understand that I'm going to say a cliche, but for me, like coming out of school, I was very much feeling like, gotta get a job, gotta get a job, gotta get a job. You know, I've got, doesn't matter, you know, not quite doesn't matter what the job is, but like going into every interview, like I'm the low person, and I just have to convince them I'm right for this job. But I think it's really important to think about whether you want the job, like the jobs themselves, like go into these interviews with the idea of talk to the people about what your daily job is, where you're going to be able to grow, where you're going to be able to develop, because then you get one that shows that you're thinking about it and that you didn't just respond to every single thing, but you actually like want this job. And number two, that it allows you to get a sense of, do I want to do this, or am I gonna in a year's time feel like, well, this sucks and I now have to go find a new job. So you got to go in with the idea that you're interviewing them as well to say, is this a team? Is this a job I want to work for?
Yeah, I think that's a great point. I'd add maybe one contrasting point, you know, between maybe more growth in industry versus growth in academia, is that I really want to deepen my technical expertise, right? And I'd imagine people in this room have that same desire. I think the truth is, in industry, like deep technical expertise isn't necessarily the point of the realm, right? In a lot of companies, managing people is much more rewarded. Might not be true in everywhere, but managing people is what's more rewarded, and you have to want to grow yourself and your technical skills. And yeah, the most technically proficient person, or technically skilled person, is I mean, academia isn't necessarily a pure purely like that either, but yeah, there's other skill sets that are highly rewarded in industry other than just increasing your technical skills. Do you guys see that, Mary?
I do, but I'm trying to change that, to be perfectly honest. Like I just telling you guys a little.
It's there.
No, it's incredibly there. It's 100% there. But like I've been like, it is the general plan of industry is get really good, impress your bosses, be good at your thing, then get people under you to train them to be good at their thing, and then sort of keep growing and developing until you are running more and more things. I'm trying to do a different path. I've never really wanted to run people, to be perfectly honest. I like the technical side. So I've been trying to go more of a pure technical viewpoint while still having broad impact in the company. It's not the typical, as Kevin said, it's not the typical pathway, but so far my bosses have been open to it. So we'll see how it goes. But I mean, I think that there's been, I think particularly with COVID, there's been a shift of what it means to work and how it means to work. And it seems like they're more willing to take these sort of odd pathways and it might, like you said, being able to manage people is massively rewarded, but it may not be the only way.
Yeah, I totally, it's worth thinking about when if you're like interviewing at a company, right? Ask them what's
How can I grow?
yeah, exactly. How can I grow? And like, is that really are the most, is that something that's actually rewarded at the company?
Yeah, because if there's no ways to grow in like the short term, right? Like 1, 2, 3 years, then it's like, wait, I have to be here for 10 years before I can like either like, change title or make more money or like whatever it is. Like, yeah, I think it's really worthwhile to understand like how you can move and grow, right?
So a very, I guess, practical question for people that are in this audience, PhDs, looking to get their PhD, graduate, and are considering industry. Do you all suggest, or did you do a postdoc before going to industry, or did you just go straight after graduation? Like, would you recommend a postdoc as a way of like getting extra skills before moving to industry, or what's your?
I would absolutely not recommend getting a postdoc before moving to industry.
All right.
Like, I mean, to be perfectly honest, like if you are dead set and you're sure you want to do industry, do it, like look for an industrial postdoc. Like if you are sure you want to go that route, because we see a lot of resumes of people who've done postdocs, and there's always the question of, okay, they've done they went undergrad, they went to grad, they went grad, so they did the postdoc. Can this person even function in an industry setting? And there's no real way to know. Did they have they done? We want to see some indication that they wanted to be in industry and that they can function in an industry setting. So that means a collaboration with a company, an internship, or an industrial postdoc. And if you just have sort of undergrad to grad to postdoc, it's not, there's nothing in there saying that you really wanted industry. Did you actually go into industry, or did you just not get the academic position you really wanted, and you're going to be, not happy in industry? Yeah. You always want academia anyway. So it's tough. Personally, I wouldn't recommend a postdoc. If you're going to do one, try and do an industrial.
I have a follow-up question, but I want to give those two a chance to.
Go ahead. I didn't even get a PhD, so sort of same vein.
Britain, I think, has a compelling point, especially probably the way the hiring market is right now. I mean, PhDs, it's always kind of harder to hire at that level, but if you're, and this wasn't the situation with me, but if you're at the end of your PhD and you don't want to do what you've been doing for 6 years because you're kind of sick of it, and you need to find a way to get different skills in order to bridge to industry, that could be a way to do it. But I think Britain's idea about an industrial postdoc might be a decent option there. I don't really have strong feelings on it.
Well, I guess another follow-up and then I'll ask that.
Yeah.
It's very interesting to hear you say that, person, because there was a time when in pharma, not in the device industry or in other careers, but in pharma and small pharma, and some biotech, where there was a clear difference between people who were hired without postdoc experience and those who were. It wasn't that everyone had postdoc experience, but what would happen is people without postdoc experience would sort of hit the glass ceiling. And the people who ended up being sort of in the higher up jobs tended to be the ones that had postdoc experience. But I don't think this is, I think your experience is a reflection of change.
Yeah.
But I don't think it's just a change about wondering what people's motivations are. If they think, industry knows now better than ever that, most PhDs are going to work in industry, and there aren't enough academic jobs. If there were, then I'd only be able to train an average of like 1 PhD student a lifetime, right? So I'm already exceeded that by So I think industry knows that there's lots of PhDs who are going to work in industry, and in fact, it's sort of a characteristic of the US industry, unlike the rest of the world for decades, that PhDs went into industry, and then they even became titans of industry. So I think that's an incentive difference that companies now appreciate. But I think the thing is, they're also looking at the kind of experience you have. And if the experience you have is doing very sort of basic theoretical science, very monodisciplinary, then they're likely to worry.
Yeah.
Right. But graduate programs like this, and honestly, most of the graduate programs at UCSD, where we've been very interdisciplinary for a long time, don't train students like that. And so I do think that one of the reasons you got your job when everyone else was applying at a postdoc wasn't because they were worried that the other postdocs, that the other applicants were all, just academics. It was because the nature of the training that you had was actually more interdisciplinary. That you were better qualified to do that job. So what it means now is that you don't necessarily need to do a postdoc, but it could be that you can still benefit from doing that. Particularly if you're not exactly sure what kind of industry or kind of applied research you're going to do.
No, yeah, that makes sense. And I agree with you. I think that particularly, like, just to give you guys a bit of my background is, I work for Andrew, and then I also worked with Susan Taylor, who's in the biochemistry department, and having that biochemistry side as well for a company like Pfizer, which does so much small molecule and chemistry. And so I could talk to the chemists and I could talk to the biologists. So deep and strong, and she's gotten enough skills, and that she didn't face that type of glass ceiling, and that has been sort of the shift, I think, in general in industry, that they've been much more receptive to different paths to skill sets, rather than it has to be undergrad, then grad, then postdoc.
It's very interesting.
Well, perhaps this is going to be kind of a moot question if, if industry is becoming a little bit more open to alternative paths. But my question was going to be, how do you market yourself to industry if you're coming out of a PhD or coming out with a master's or something? So at least, like, speaking for my home program, we're starting up a program where PhD students can leave for a summer to go get industry experience and then come back, finish their PhD, and then maybe even go back to the same industry position after. Is that something did either of you or any of you participate in that, or maybe an industry postdoc?
I've seen that from the other side. Basically, we've had interns and things that we've worked with. I think it's beneficial if you know that's the industry you want to go into.
Okay.
So, like, if you know you want to work for big pharma or something like that, then doing that at a company like Takeda or a company like Pfizer or a company like Amgen makes sense, because then if you're going, once you go out, having that on your resume really is. I definitely, like, for me, when I see those resumes, I put those in an additional tier. I sort of put a little tick box going, okay, this person has this skill that I'm looking for.
Probably it's pretty hard during a degree program to demonstrate interest in industry.
It can. No, 100%. It really is, and that is always the challenge. And it's the challenge for us, is because we don't. When we're looking at all these different candidates, we're sure many of them are very good and will fit into the things well, but we just have to. But without it, we just have to sort of figure out in the interview, and then a lot more sort of relies on how you did in your interview. Were you able to answer the questions well? Were you able to show that you could collaborate with others well? So it's a lot more like hard on that interview if you don't have something like that where we can go, okay, we know they have the skill.
Yeah, that's a good point.
I think in terms of marketing yourself, like beyond just like an internship or like specific programs or, like some defined action. I think it's like a little cynical, but it's really, really, really important to use like your network of people to market yourself. Like it's an unfortunate reality, but it is like extremely true that like use the people who you're in the lab with, like use your PI to help you as much as possible. Cause it's like submitting yourself through like a job portal, in my experience, is fairly fraught. Like being able to say, I know someone who works there. Hey, can I talk to you? Blah, 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 blah. And then all of a sudden you're like 2 steps ahead of someone else. Like that to me is like the most important thing I would try to impress upon you is like, yeah, there are these like discrete activities you can do to like pad your resume, but like working your network of people is, in my opinion, like the most surefire way to give yourself a leg up in any of these situations. What do you all think?
It's unfortunate, but it's 100% true.
Yeah.
It's a completely 100% true.
Yeah, it's both. That's not how I ended up with my position.
He's a good
In fact,
guy.
when I tried that and I kind of failed. But
I think also when you're at like a
True.
larger company, perhaps that probably helps a little bit more too. Maybe there's like more chains of command you have to, impress to get the job. So I can only assume.
Mhm.
Okay, all right, yeah, this is kind of all along the same line. So this is another question about what PhD students should be aiming for. And I guess the question is kind of related to publications. So academia, we're all like led to believe academia, it really matters the impact of publications, how many of them, and then, trying to climb up the ladder. But does industry, when you look at a resume, appreciate, the impact factor of a publication, or how many publications? Do they even matter, or do skills matter more that you can demonstrate? Like kind of what's the process there? What's the thought process for someone who's more bound for industry in their head?
I mean, I would say that the skills that person has and their ability to solve problems would be more important. I don't want to say that too loud, maybe, but I mean, definitely, a publication, somebody who's coming out of PhD, you're going to look at their publication record. Like it's important, it's a factor. You want to look and see what they did. And you also want to look what part of the papers did they actually do, right? That's a conversation that I've had with people coming in. Like, I want to know, what on the paper they were responsible for. So even being on a big name publication where you had a very small role, which is important in academic life, of course, I'm going to know like what skill you contributed.
Yeah, I think also like the relevancy of the publication to the position, right? If it's like some super sick publication, but it's like tangentially related to like what the company does, then it's less attractive than something that's really specific and super relevant. Because then if you're trying to hire this person, it's like, wow, they could have an impact and like move us forward right away, right? Like they can like give us so much expertise. I don't know. What do you think?
I think the publications might get you in the door, to be perfectly honest. Like if it looks good, like if you've got a decent chunk of publications and you've got and they're in a relevant area, then that's honestly impact factor. I don't look at impact factor when I do it. I just look at, what the names on their publications, what they did, things like that. But for me, it's much more of anyone we bring in, they then have to come and talk about their research. They have to give an hour-long talk about their research, and they have to be able to know it. And we don't want you to say like, well, why'd you do this? Well, my PI told me to. Okay, well, see ya, next person. It's much more, it's much more, how did you move it forward? What skills did you use to do this? How did you show your impact on this that you can then bring to the company? And if you can talk and take questions about it and sound intelligent, and even like, I love the people who like go, here's what I did. Here's what I wish I could have done. Like, I didn't get this done, but here's what I wish I could have done. I would have done it like this. I would have done these controls because I really think that shows that you have the mental capacity to think even beyond what you've already done. So personally, it's so much more that than your publication record.
That's a really awesome answer. Thank you all so much. Yeah, it's super cool.
I think just switching gears a little bit to a different topic, a harder topic perhaps.
Ooh.
What are some of the most challenging parts of your job that you potentially wish you could change? Good for us to know what we're getting into if we go.
Timelines. You a lot of times, a lot of times when you do what you, in particularly in industry, it's do everything fast, and it's every program is the most important program. And so that can a lot of times make you operate on a timeline you don't want to operate on. So basically, you'd want to do this, 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 and this, and this to really make sure everything is exactly right, but you only have this amount of time, so you have to do this. So for me, I would say timelines is the hardest part, is to basically be able to deliver quickly and with confidence and with knowledge when you might not have all of it.
Really the opposite of what scientists tend to do, academic scientists.
True. I mean, yeah, I mean, but that's it. And you have to be okay with that. And then at the end of the day, you have to be okay with where you ended up with that.
Yeah, and I think having the understanding that like some things won't get done sometimes,
Yeah.
right? And then it's like, how do I prioritize these things? What's the most important? And then kind of like adding on, definitely yes to that. And then another thing that I don't know if it's challenging, it maybe stresses me out the most is like sort of like not necessarily making a mistake, but like maybe in a situation where you're too slow or something doesn't go right, like how that then impacts your coworkers, right? Because it's like you really are teammates, and it's less of like a solo activity than, you know, academic research. And so it's like, oh shoot, if I mess something up and then this thing's delayed by a week, it can have like, you know, like outsized effects, and then it's like impacting other people's ability to do their job. And so that can be really, really stressful in some ways, right? Because it's like you want everyone to succeed, and then when you make a mistake, it can be pretty bad.
I would agree with both those 2 points. You have this in academia also to some degree, but you are reliant on other, you know, like I really, like I said earlier, being collaborative is like one of my favorite parts of my job. Sometimes you're reliant on other team members that, you know, come from that can be a challenge, right? Sometimes I work with a great team. Every once in a while, you end up, your ability to deliver something is kind of requires you to work with more challenging people than maybe you would sometimes come across in academia. And that's it's a new situation for me to deal with, and kind of figuring that out and handling that, you know, kind of professionally and in a way that didn't impact timelines and still allowed delivering the quality work that I wanted to deliver that was a challenge. That was hard.
No, I really like, I hear that a lot. Like, I completely, like, and not to stereotype a little bit, but like there's a few more aggressive people in the industry. Like they play the political game. They are more sort of upfront and very aggressive personalities. Not to say there aren't aggressive personalities in industry or in academia as well. We've all been at conferences like that. But it's definitely working to manage them and working to manage your relationships with them, because you might need them still and you have to work with them. It can definitely be, has definitely kept me up at night sometimes.
Oh yeah.
In terms of,
I have a follow-up question to that
Okay.
question. Can I do it really quick?
yeah, that,
Cool. Okay. So I've spoken with like just a bunch of my friends, just my cohort, you know, and everyone's at this transition stage trying to figure out what they want, right? And I think I hear very often, and I want to confirm, is it true that there is more work-life balance, or that it's appreciated more in industry? That seems to be something that constantly comes up when I ask a graduate student what appeals to them about it. And I just wanted to know if that's like a rumor, you know, or is it like people that always are workaholics are continuing to do so, you know? Is it just the same hogwash it would be in a graduate program? I just wanted to know, like, what does it look like, you know, just around, you know, different companies?
Can I take this one? I think the work-life balance is better, but I think it's because most grad students don't understand their value and therefore feel bad taking time off.
Yeah.
Like I have a friend who's still in his PhD who he takes a lot of vacation and it's fine and his research goes swimmingly and it's not a problem. But I think when you're in industry, because there's like these like, you know, oh, you have unlimited vacation or you have this much time, and it's like, I don't even know if you're working more, but you're like working and it's maybe a little more structured. And then you kind of have this notion that it's like, great, like I'm working, I'm earning, I like deserve this time off. Whereas like, I think in grad school, like it tends to be this kind of like inversion where it's like, oh, I'm like, need to like impress my PI, like I can't take this time off, I'm like so lazy, yada, yada. And so I think it's just like maybe more a case of people not understanding their worth in grad school as opposed to it explicitly being better in industry. I don't know, I'm curious what you guys think.
I think, so let me take the question from a side direction and then go back to it, which is like, what you said is so true, which is, and functionally, I apologize, we're gonna talk about money for a second, but like, you get paid in the industry. And so there have been multiple times when I've gone to my boss and said, hey, you pay me this much. Do you really want me spending my time this way? Like doing this grunt work? And the answer is no, we'd rather pay a contractor a third of what you get paid to do it while you focus on the most important things. And so really speaking to that value piece, because they're paying you what they're paying you, they have to value your time. And so therefore it makes it so that, do you never do grunt work? Of course, every job has some grunt work in it. But at least you can go to your bosses and say, this is my time being spent correctly based on what you're paying me to do this. On the work-life balance thing, COVID screwed it. I mean, I would have said, if you had asked me before COVID, I would have said it was a good work-life balance. I was really happy with that. COVID has done a lot of things to a lot of things. My work-life balance hasn't been great recently, to be perfectly honest. But I think that there's definitely more of a feeling of almost everyone I work with is a family person. And so everyone understands the value that requirement that has children, has a wife, has a husband, and therefore there is a mandate to even if you're working really, like if you're killing yourself these 3 months, then you got to take some, like, then maybe the next 3 months you need some time off, take a breather, take a rest, you know. So I think it's better, but I mean, I've never worked as a professor, so I don't know how it is there.
We get great vacation.
I'll give you a chance to answer.
Sure. I mean,
if not, that's
I
okay.
don't have too much to add. I mean, it's up to you. I mean, I work at a much younger company than Britain, probably on average, and there are people that work, people in their mid 20s, late 20s. Some people really like working hard, and definitely there are people in my company that spend a ton of hours. But if you have to advocate for your own work-life balance, I think. Like, you have to set your priorities. You have to be efficient, and you can have that certainly in industry. Just maybe it's harder in academia, but you can definitely have it.
Sure, it also varies job to job a
Sure,
lot,
and
yeah.
depending on the size of your company also, like the big company like this might be a lot different.
The big ones I think are actually better on the work-life balance. The smaller ones, like particularly if you go for a startup, like just what I've heard from my friends who work for startup, it's not so much it's an eye on you, it's that you have a 6-month runway.
Yeah.
Get it done, or the company's folding, and so therefore there's a lot more like, okay, you've got to put in whatever hours required to keep it going. So that's just what I've heard. I've never worked for a startup.
So do you have another follow-up?
No, that was it. Yeah,
Okay,
thank you so much.
My next question was, so we talked a little bit about the challenges, working with some people that maybe you would prefer not to work with, having some, some hard and fast deadlines, and kind of just adapting to like the industry lifestyle. What's mentorship look like in industry? So do you kind of learn that? Do you learn how to adapt, super quickly in the first like 3 to 6 months in kind of a sink or swim environment? Or do you have people that you feel like you can go to that can help guide you for how to adapt? What do you think?
Sure. I think it's harder and somewhat harder in industry, at least in my experience. My company doesn't have a lot of other people that do the type of work that I do. So finding the people that I want to mentor me has been, it's been a little bit of work. I feel fortunate. There are some guys that aren't in our office that are like a neurologist and a neurophysiologist that I've kind of, leaned on for that type of mentorship. I've also enjoyed having young teammates that I have the ability to mentor. That's been a part of my current position that I have valued a lot. But yeah, I mean, there aren't as many formal mentors available to you. You have to kind of find your own. I don't know what you guys think.
Yeah, I think it depends on the company. Like some places I've worked, there was like, hey, out of these 5 people, pick who you want to be your mentor, and then you all have like dedicated time together. And I think that's like really cool. In my current gig, that's not the case. But I think it's kind of one of those, like, if you reach out to them and say like, hey, I think your career is awesome. What you do now is totally what I want to be doing in 20 years. No one's gonna be like, go away, right? Like, they'll be like, wow, that's so flattering. How can I help you, right? And I think it kind of is on you as an individual to speak up a little bit and try to get that. I think mentorship can kind of mean a lot in this situation. Like there's also kind of the idea of like a manager, right? Where it's like, oh, if you're having more discrete problems, then there is someone that you can go to like have them help you, but kind of like big picture, capital M mentor, maybe you need to seek out a little bit.
Yeah, I would agree with both of them. And I think that I would say for this particular question, there isn't an answer for industry. I think it's every job will be different. Every position will be different. So find a good position where you feel like you can be mentored. Because again, just from friends I've had, personally, I feel like I have had a good mentorship, but I've had friends who've said exactly what you said. It's hard to get, it's hard to have, it's, you know, they don't feel like there is that position in place. Yeah.
I think,
So,
sorry, I was gonna say, I think also if you consider a mentor someone that you're just like learning from, right? Like any teammate, someone who's like on your same level, even below you, like they have certain things to teach you in certain areas, right? And so it's like, if you're on a particular project with someone, right? Like you can definitely learn from them, right? And I think being able to absorb that and like really embrace that is super important as well.
and I don't think it's as much sink or swim as you said, sort of. But like the learning curve is huge in the first 6 months. But I never felt like it was like, and therefore I'm going to fail. It was much more of like, the learning curve is huge, so we're going to learn. You know, we understand, and so I was sort of more eased into like, I didn't have what I would consider now a full workload for at least a year. My first year working there, typically I have something like 5 to 8 projects. I had 2 for the first year, and then it got to 3, then it was 5, you know, and sort of built it up from there.
And every company knows that, right? Like every business knows it takes you like 6 months to a year to ramp up.
Yeah.
Okay.
And it's like the expectation is like, you know, you don't sit down on day 1 and they're like, great, solve this problem, right? It's like, here's how you log in, right? So it's like,
And if they do, you're at a bad company.
exactly. So they want you to succeed.
Kevin had more of a just dumb.
Yeah, here you go.
The day I showed up to work, and then I had like the little HR speed, you know, like they told me all the benefits, and then they were like, all right, here comes your manager, Ed. And I was like, who's Ed? The guy who hired me and quit before I started. And it was, I came into like a crazy situation. It was very like tense, and it was like, I don't know, like the CEO came to the company, the company came to a meeting, was like, don't fuck it up. Excuse my language. The vulgarity is a thing in my company. But yeah, so that was like a thing. But I've been very happy with, you know, I had to solve a problem. I laid out how I was going to solve the problem, and I executed to that plan that I laid out, and I've been rewarded for it. So like that was it didn't have 6 months in. I had a machine learning algorithm in the operating room that was that was being used. I was not like coming up to speed, but yeah. So it was a different on ramp for me.
Kevin took off further.
Yeah, you know. But sometimes you got to take that opportunity. You never know where you're going to end up. So when there's opportunity, take it.
Well, it's nice to hear that industry generally is always very encouraging of, you know, growth, and it's not as sink or swim as I said earlier. So that's nice to hear.
Are we coming up?
Yeah, what
Maybe
time
the
is it?
last few questions. This is a little ahead, just to have me in places on time.
Oh, okay. Yeah, we could skip the last one and then ask all these.
Yeah. So, okay, our last question, which is going to be different for all of you, doing different things, is what's changing in industry, and are there any future trends that we should all be aware of?
Or what changing in your industry?
Yeah, in your industry specifically, or that you know of more broadly. Just
Everybody's becoming a data scientist. That term means basically nothing. No, that's
No,
probably
I think you're absolutely right about that. I wish we had more of it.
Sorry.
I would say the big change has been COVID. in a lot of ways. And so I think I was telling some people at lunch, it's sort of like I'm getting a lot more offers for job postings where they're like, hey, we're out of Maine or Idaho, sorry. But we're fine with you working 100% remote. So you don't have to move. So if there is companies willing to do that, that might open up your prospects when you're looking. So don't necessarily just keep yourself to San Diego if you want to stay in San Diego or something when you graduate. Think about some of these remote positions and look to see if there are some that you, and whether you want to do something like that.
Yeah, I totally agree. I worked remote prior to the pandemic, and it's like, even then and way more so now, right? Like those opportunities exist, especially if you do something computational where you can just, be click-clacking away all day. I do think like in terms of trends, it's like, it's very challenging or it's worth being mindful of like how you kind of separate your work from your life if you are working from home in that capacity, right? And you need to like really set those boundaries and like try to be as healthy as possible, because like, in a more traditional sense, like commuting to work, like that's a good transition time when you can like change your mind, right? And kind of like ramp up and then ramp down. And that can oftentimes be really hard for people when you roll out of bed and you're straight on your computer on Slack. Like that's horrible for your mental health. So something to keep in mind.
100%. And people are, at least my experience has been, people are much more willing to email you off after hours now with COVID, like because everyone's working at home. And they're like, oh, well, I'll just shoot off this email at 10 and expect a response. And it's like, all right, well, now I have to be checking my email all night. And that's definitely really bad for your mental health. So it's definitely something you have to sort of manage.
Well, I think we have a couple more minutes left. Are there any questions from the floor before we close up? What's your favorite part of your job? What do you enjoy the most?
I'll jump in. So I really like working with the people in the field who are using the stuff that I've developed and hearing feedback from them and then proposing my ideas to them and like getting feedback with people who are actually using stuff, in the operating room with patients. I love that part of my job.
I would say something kind of similar. It's like when let's say I'm working on some particular tooling or, like a model to like surface some biological data. And then when I present it to the biologists on our team and they're like, wow, that makes complete sense. That's like 100% right. That like satisfies all of the things we were thinking would happen in this experiment. It's like, that's awesome, right? It's like things work from multiple angles is extraordinarily satisfying.
Yeah, exactly what Kevin said is like, and having stuff in the clinic, having stuff helping patients' lives, like being like, hey, we developed this and these people are living longer because of it. Like that, the placebo had this effect, you had this effect, these people lived an extra whatever amount of time because of it. Like, I can't tell you how amazing that. Like that's my favorite part.
For Britain, it's like it's real. Those are the COVID antiviral drugs, right?
Yeah, wow.
We've done some pretty incredible work there. You have a lot to be proud of.
But that's what we do this for. Like that's why we went into this, is to do that, and it's awesome.
So it's maybe a kind of a weird question and maybe more personal, but like, where do you see yourself in 5 years? Like, do you think you'd be in the same company? Do you think your role is going to change significantly? I don't know. I feel like as people that are looking to get in the industry, we could ask that in an interview, right? But it'd be interesting to hear people that are already in the industry, kind of what that might look like.
So what do you guys want to think?
I'll probably be at the same company. It would be my guess. Like, I'll probably be in more of a senior position, but basically doing what I'm doing, just doing it at a broader scale.
Yeah, I really enjoy the place I work now. And I think that's something kind of reassuring, right? It's like I enjoy where I'm at, so there's not really a reason why I would want to jump ship for any reason. I mean, ideally I'd be in more of like a leadership position, blah, 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 right? Like, there's a variety of ways and directions in which you can grow, like we've discussed, but like broadly when it's like, oh, things are going great here, like they probably will keep going great. Like, so why rock the boat?
I would say something similar. I would like to be at the same company and kind of pursuing the type of path that Britton described earlier of, getting rewarded for technical expertise as opposed to like growing more towards a manager. But, 5 years seems like a long time.
I was going to ask if you believe that the traditional bench scientist role is dying out in the industry, if there's more focus on computational methods, or would you say you can still drive just the bench side?
We hire both. I think that bench scientist is somewhat of a misnomer, maybe, because like if you're thinking you're just going to be pipetting all day, then sure that role exists, but it's not well endorsed. And honestly, they don't want a PhD candidate for that. They want an undergrad or someone with a master's with that. Like you can be a bench scientist, but your job might be to run 15 different types of experiments. And like basically your job is to design the experiments and make sure that they're run correctly and then analyze the data afterwards and deliver it to a team of what this data means and help design the next step. So I mean, I don't think it's I think bench scientist is a bit of a misnomer. I think it's much more experimentalist, 100%, but you have to be able to sort of be more of a overseer of experimentalists as well.
There's not a lot of there's a little bit of biologics work at my company, but there's not a lot of bench scientist work in my company, at least. I can't really speak to the broader industry.
Yeah, I don't know. I don't do bench science work, but like we have a few at my company. One thing I would say about computational work is that if you want to pull an insane pivot and like go work at a pure tech company, those skills are more transferable, right? So with computational skill sets, you probably can work in a broader variety of companies. Maybe not. Again, I'm not a bench scientist, but that is my assumption.
Any other questions?
I can add a further perspective that in pharma, what you need is data. So, you think your lab gets a lot of data? Go to a pharma lab. They really get a lot of data. And data, the quality of the data, the controls and the design experiments, all very, very good. And that's why they need PhDs too, because these experiments they spend a lot of money. Now, today, as opposed to 15 years ago, the big difference is it's much more likely that a lot of that data is actually acquired somewhere else.
Mm-hmm.
Particularly in a smaller company, it's a very good chance that it's contracted out to a CRO, possibly on the other side of the world.
Yep.
That doesn't solve all problems by far, because we still need to design as if anything makes it harder, right? If it's hard to oversee an experiment that's done in your own building, I go to see an experiment that's done, halfway around the world by people who don't speak the language. So data is more important than ever, and data comes from doing experiments. It's a lot more of it, which drives a lot more data analysis, which then produces a whole lot more data. But you can't make data out of nothing. So, all the computational models in there and the data science still needs to be driven by actual experimental results, actual clinical observations. But who's doing them and where they're being done, that's a much more complicated question now. But despite that, around 800 companies right around the data, there is, almost all of them people doing it. Literally all of them, but most of them.
I'd say I probably spend about 25% of my time just designing experiments, just designing what they like, working with a biologist, working with a chemist, so that we have the different pieces of knowledge base there, and designing the best experiment to answer the question to move the project forward.
But I guess those experiments are expensive, so it's not like when your place says, "Oh, why don't you do this experiment?" This is the experiment that we're designing. It's going to cost $150,000.
Yeah, that's right.
And they want it done.
Yeah.
Awesome. Cool. Well, I think, is there another one? One more question? Anybody else? Okay, I think we're gonna call it there. So thank you so much for your time.
Yeah, thank you
much.
guys.
Thank you guys for your insights. That was really awesome.
Nice to meet you guys.
Yeah.
You look really cool.
All right, so we're gonna take a break and we'll be back here at 2:45 at the academic. Thanks, guys.
Yeah. I bet so.
For drug delivery and sensors for imaging in the brain.
Hi everyone, my name is Itai Budin. I'm an assistant professor in chemistry and biochemistry, as well as bioengineering. And I started UCSD in 2019. My background, I was actually a bioengineer as an undergrad at Cornell. And then I actually did my PhD in biophysics at Harvard. I worked in a lab studying lipid membrane. I got really interested in lipids and cell membranes. I still work on today. I did a postdoc in Berkeley as part of a Miller Fellowship, which is kind of an independent funded postdoctoral fellowship thing. And I worked in a lab there that actually had a lot of people that went to industry or started companies while I was there. A lab that did a lot of work that was relevant to synthetic biology and biotechnology. Academics, yeah, then I started my lab here in 2019.
Hi everyone, my name is Ernesto Criado Hidalgo. It's the other way around. My background, I'm originally an aerospace engineer. I did aerospace engineering back in Spain. Then I actually worked in industry for 3 years in a startup where I was working, developing explosive trace detectors based on ion mobility mass spectrometry. And that's when I started kind of slowly shifting more towards biology. I came to UC San Diego. I did my PhD here in biomechanics with Juan Las Heras and Juan Carlos del Álamo. And I mostly worked on 2 different projects. So one was more dealing with typical biophysics problems, cell migration, quantifying mechanical forces in cell migration. The other one was more related to my original background because it was more fluid mechanics. And I was trying to understand the dynamics of the cerebrospinal fluid in the spinal canal. And now I'm a postdoc at Caltech. I've been for a little over 1 year there with postdoctoral fellowship, and I'm working with another Miller fellow, Michael Shapiro. I'm working right now on developing genetically encodable tools to leverage mechanical forces to control cellular function.
Cool, thank you guys. So our first question is, or actually, we want to ask the audience if you have any burning questions before we start, and then we'll give you an opportunity to ask questions after we ask ours. Okay, so our first question is, what prompted you to pursue a career in academia? I'm sure you were sitting there as a PhD student at some point and decided to go do that postdoc, or from postdoc apply for that professorship.
Sure. My intention was actually always to go industry when I started graduate school. I did 2 internships. I did one between right after I graduated, and then I did a second one while I was a grad student actually at Seattle Genetics, which is a monoclonal antibody drug therapy company. I really enjoyed my time in industry in both instances, but I think the thing that switched for me between my 4th and 5th year of PhD is that I really loved the structured mentorship in academia. The fact that you can help a student develop over like in a very formalized way, like they come in, they do their coursework, then they do their qualifying exam, and then they mature, and then they have their own ideas that they pursue into their general exam, which is like our Senate exam, and then they defend. I really like to see a student grow on that trajectory, and it's very formalized in academia, so I love that component. And then the second thing that I really liked was the ability to pursue like my own little crazy random idea and pursue that to the end. I also really enjoyed that aspect of when I was as a PhD student. So that's when I decided, basically in my last year of my PhD, that I would try a postdoc and see how it felt and how it fit. And that's how I decided.
I mean, I would say like growing up, I definitely didn't say I was going to be a professor or anything like that. I think, as Esther said, you're doing your PhD and you're kind of seeing what things about your work you like and what things you don't like. And as for me, I really love being extremely independent as a PhD student. I loved writing papers. I loved giving presentations. And so to me, those things, into academics. Yeah, I also love teaching as a PhD students, of course, that's something structured way and formal way. So,
I would say, well, I'm still a postdoc, so I would say there's still hope for me. But I think in my case, when I started working for this company back in Spain, so it was a startup, and one of the owners was actually a professor at Yale University. So then we had a lot of interaction with him, and it was very stimulating. So we were working in industry. We definitely were subjected to those timelines, and we had to develop things. We had to prototype fast. We had to do a lot of things, but it was also very scientific at the same time. And actually, I mean, we got to publish several papers from our work there. And that's how I got interested into, hey, maybe, doing a PhD is not such a bad idea. And actually, I got some exposure because I got to work with this professor for a year before I started my PhD. And then, I mean, the way I still see things right now, I've been fortunate enough to have some industry experience. Right now, I'm working in a lab that kind of functions as a small company. So there's definitely a lot of strict timelines. You have to report I would say pretty much every week. So there's a lot of things. You have experts in different fields. Like if you need some cryo-EM experiment, you talk to the cryo-EM guy. If you need some, I don't know, more protein engineering thing, you talk to the protein engineering guy. So I think it's, and that's the way I see things. I mean, obviously it's not every lab is like that, but there's definitely that experience where the academic world can also be a little bit geared towards developing things and kind of making the most out of everything.
So just a follow-up question, are you planning to go into industry after, since your lab's kind of focused like that, or?
My plan right now is to pursue an academic career after this, because I kind of enjoy the freedom that you have as a professor and the possibility of pursuing your own ideas and your own enterprises.
That seems like a common theme among the three of you guys, because you really enjoy the freedom that you get as a faculty member.
Okay, our next question is, what is the interview, like job hunt process like to become a professor?
Let's go.
Yeah.
Well, I can start, and then we move it up. I think, in my experience, finding a postdoc is relatively easy. Finding the right postdoc is a lot harder. And but with the kind of skills that we get as PhD graduates, you're always going to find someone that's going to hire you to do several things. But I think it's important to think of, like I always think of this advice that I heard somewhere. I mean, take your job always thinking of the next one, right? And that's when it comes to, well, if you want to be a faculty member, then what kind of postdoc do you want to do? Because you kind of want to, at the end of the day, when you get into the academic market, you need to tell a story, and you need to tell a story that makes perfect sense and that everybody loves, even if it was everything but perfect. But that's your ability as a, I mean, at the end of the day, I think academics, you also need to be a storyteller, and you need to be able to get things from here, and then come up with something well rounded up. So I think it's worth taking some time to explore whatever postdocs or whatever labs you may want to or you are considering. Talk to people, talk to current members, talk to former members, see where they are, and then be flexible, because a lot of times, and then I mean I can speak from my own experience, things don't work out. I mean, the lab that I'm currently in, I never in my life planned that I was going to be working in this lab, but it was just, I got a fellowship, a postdoctoral fellowship. The minute that I arrived at Caltech, the person I was going to be working with left. So then I was, and this is summer 2020. So it's like pandemic, so it was a complete mess, but everything turned out great. And I'm super happy being where I am.
Yeah, I think choosing your postdoc is very important. And I would say, like, the postdoc one that I chose, I don't think was necessarily the ideal one for my project. I came out of grad school very naive, thinking I could do my work anywhere that had the right machines, right resources. And actually, I think that was not true. And you really need to think about like, where you want to go and what group will take you there. Because really, go on the academic job market, it's really going to be your postdoc. In most cases, it'll be your postdoctoral work that people are evaluating or basing their hiring on. That is going to be what your namesake. So it's a really important step. Yeah.
Yeah. So when you start your faculty position, you'll need to be able to verbalize succinctly why you're distinct from everybody else, like everyone who's doing research. So that's why choosing your postdoc and your grad student, your PhD, and having those complementary and how they work together and how they will propel you to this new area that's distinct is important. I don't know if the question is more about the nuts and bolts or like, but
More philosophical,
philosophical.
more on your experience.
Yeah, and then I think when looking for the right environment for your future, looking at not just your own home department and the strength and quality of that, but also the departments that, as you guys are all interfaces, right? So you know that we all work together. So finding the types of expertise and whether it be clinical or if you work with a lot of material scientists or physicists, making sure also that's strong in the future home department that you'll be going in, knowing that you'll want to take students from those different areas in your own group. So that's also really important to evaluate when choosing. For me personally, choosing UCSD was, kind of a no-brainer considering the strength of the graduate students in our program here. And so because I know that I love mentoring graduate students and my lab is going to be mostly graduate students, for me, that was like the #1 decision to find a program that had the best graduate students. So that's basically kind of how I chose. But I mean, the nuts and bolts have a lot to do though. Like that's like, ideal, and there's like reality, right? So like I applied to 40 universities and I interviewed at 8. And you get the offer that you get the offer in. And so, and then you choose the ones that you like the most. So those are like, that's the, reality meeting expectations. So, but when I had time to choose that, it was based on the students for me. And then the neighboring departments.
Cool. So as a professor, you all have roles as both educators and researchers. Do you see yourselves leaning more in one direction or the other? And then what made you decide to pursue a mixed role as a professor versus a role as a lecturer and just as an educator?
I mean, I'll just say, your primary job at a research university is research. You also have to teach classes, but I will say the research, you're also an educator in research, because Esther says you're mostly working with grad students or postdocs who are trainees as well, and you're teaching them how to do research. You're also learning yourself all the time as well. So I would say, like, the educator is not like you're in the classroom and you're an educator and then you're in your lab and you're a researcher. They really kind of blur.
Yeah.
And that's not necessarily true, but I think that's almost true in sort of academics.
Yeah, I would definitely agree with that. And I think it kind of happens at all levels. Because when you're a graduate student, you're doing your research, but at the same time, you're mentoring other, mostly undergrads or even other like, less senior grad students. When you're a postdoc, kind of the same. I mean, you have undergrads, you have other rotation students and things like that. So I think it's always complementary. It kind of, I mean, in my case, I love doing research, and that's the most important component for me. But at the same time, the teaching part, it sort of intertwines very well, because you need to be able to explain things as a researcher. And sometimes explaining things to other people is the best way for yourself to truly understand what you're talking about. So I think they kind of, both things come hand in hand.
I agree with all those things. Just wanted to add that there are different types of universities. There are large public universities like ours, where your classes will be 100 to 200 undergraduate students. There'll be smaller private schools where your class size will be more like 50 students. And then there's like more like teaching universities where research and teaching might have an equal role. And so you may consider that depending on you know, where your goal lies, if it's more towards research or more toward teaching, and how you want to balance those things and what types of settings you might want to be teaching in. So the experience can be quite different for depending on what university you end up at. So just wanted to comment on that.
It could also be a medical school or a research institute where there's no classroom. So,
Our next question is, how do you keep up with current research papers and conferences while also pushing your own research forward and keeping up with that?
I'd say you don't. No, I mean, you try your best, but especially the times that we live in right now, there's so much research, so accessible, that I think the problem here is not, I mean, it's impossible to keep up with everything. The key is to choose your battles and really, okay, I want to focus on this, I want to focus on that. And as much as I would love to study this other problem, I just maybe at this point don't have enough bandwidth, and maybe this particular thing, I will, you know, let it rest for some time, and then maybe I'll recover it when I become a faculty or things like that. So I think it's just a matter of the way I see it. The most difficult thing is choosing the right battles that align with your main goal. Because, I mean, we have so much access to information right now that you can very easily get overwhelmed and then get lost.
Yeah, it's a really hard question. I don't have an answer, but what I try to do is pre-decide how much I should do of each thing, and then I put it in my calendar, and then I just like let my calendar tell me what to do. Because I think if I just decide day to day, like, oh, this is super important, I have to do this, like I'll end up just doing like probably one thing all week instead of doing what I'm supposed to be doing. So my postdoctoral advisor, she took like 2 weeks off every summer to retreat and like think about directions for the lab, what she wanted to accomplish that year, whether it be, you know, outreach. She was really thought she really wanted to contribute to women in science. So she would also, you know, all the things that's so important to her, she would identify and then decide how much time she wants to give to those. And then, you know, I have a running tally of things I do all year. Like whenever someone asks me to do something, I'll say, okay, I did this panel thing. And then I write down. And then I start counting, right? Like, okay, I did the things, I want to do 10 of them. So I did 10 of them. Not like now I have to say no, because otherwise I'll just be like sitting on panels all day. And then my students, you know, they don't ever see me, and that would be bad, right? So that's kind of my strategy. And then I just try to come up to surface for air and just be like, oh, am I doing what I said I would do, or decide what was important to do, and then go back down and execute basically what I had decided.
So we're not, as grad students, we're not the only ones who feel like we're drowning in papers all the time.
Yeah, exactly.
Yeah, I mean, specifically on the question of keeping up with literature and papers, I don't, at least my field is very active on Twitter.
Yeah.
And it's been a really great way to actually keep up with literature, because someone will post their, you know, preprint or paper, or that'll be retweeted, or anyways, for me, that's been like a saving grace for keeping up with literature.
I completely agree with that. I mean, I was fairly new to Twitter, and I mean, it's an amazing resource for academics, and surprising because you wouldn't really expect, but you can kind of sense the importance of certain things, the impact, and kind of follow some of the things, because you're following directly the lab who's developing these things, and you kind of get the reactions there. So it's definitely a useful resource that I wasn't using, and over the past year I started getting more into it, and you get a lot of information out of it.
As well. How did you guys choose the focus of your research programs, building off of what you did in your PhD and postdocs?
Right, I'll say like, I kind of, when I was an undergrad, I did a summer research program, and basically what I did in that summer research program was, that's been what I've done ever since, in some senses. So I think but I don't think people are born to solve a specific problem or anything, but I do think everyone's mind works a little differently, and certain research topics are better suited towards how different people are thinking. Like I always like thinking about the sort of the biophysics of molecules. So for me, studying cell membranes is very natural, because I can think about lipids moving, diffusion, these things, and I really like that. And I think you also need to think about, you have, as Esther said earlier, you have to be distinctive from everyone else. So you have to also say, okay, what kind of new insight or new angle can I provide? Where, what discipline can I provide that? What topic I can do that?
Yeah, I mean, for me, I think I followed a more non-linear path, because I remember when I had to decide what to do for college, I remember thinking, well, I really like engineering, I'm good at math, physics, I enjoy it, but I also like medicine, you know, and I was always in this decision. I didn't have any engineers or doctors in my family. And then at the time was like, well, most likely you get an engineering degree, you're going to what I did for my PhD. I worked in quantifying mechanical forces in biological systems. And then for my postdoc, it kind of, or I found a way to leverage that knowledge that I had from my PhD to say, well, we know how to quantify mechanical forces in biological systems, but this is very cool. We can plot all these cool things of cells migrating and whatnot, but can we actually use that to do something to control cellular function? And in the lab that I'm currently in, Mikhail Shapiro's lab, he actually has the answer to solve some of these problems and to use, in this case, ultrasound to apply mechanical forces to specific or to trigger specific pathways inside cells to literally control cellular function with mechanical forces. So that's kind of the non-linear path that is starting to make sense and to kind of converge into something.
Esther, do you want to add?
Sure. I have a lot of interests. I would say everything. I used to do like single molecule unfolding with laser tweezers, and I love that. And then I did like nanoparticle engineering, and I love that. And then I did neuroscience. I also love that. So I'm a person like with a lot of interest, and I like everything I have done so far. And so when I was choosing my area, I tend to like really large sandboxes to play in. So like a lot of space and not a lot of. I'm not very like want to compete with someone head to head and race. And so I tend to look for spaces where no one's doing anything and like I can kind of like tinker and fiddle around. So those are areas I'm kind of naturally drawn to because my interests are pretty broad.
So just as a follow-up, when you apply to a school, do you already have to have your research projects that your lab's going to be working on?
Yes, when you apply for faculty jobs, you propose future research projects. You usually will propose a 3 to 5 year program that you will do. It doesn't mean that you have to do them, but it's an example that you're capable of processing through that, execution of a thesis projects, fundable projects. So it's an example. But yes, you will have an area. I think I am doing what I propose to do. So I think it varies person to person, whether you actually do it.
I think it's also just to kind of build on that topic. When I was actually interviewing for my postdoc, I remember talking to my PI and kind of telling him like, well, I'm actually a little worried because I've done, like aerospace, mass spectrometry, biomechanics, I don't really know, like it's a little bit all over the place. And I mean, his first reaction was telling me, oh, I think that's great. So it also depends on where you're going and what the goal of the position you're trying to take is. And I mean, in the particular lab that I'm in right now, I mean, there's a number of things going on ranging from, like I was saying, structural biology to MRI to ultrasound, molecular imaging. So there's a number of things going on. And it's great because you get a lot of exposure to all of those. And you do see how there's a lot of projects that end up being really nice papers that kind of take parts from all of these disciplines. So I think having a diverse background is not, I mean, it's not a bad thing. You just need to put some effort into making it work and making it work together.
Okay, our next question is, how would you recommend us as PhD students connect with additional mentors besides just our PI?
I think conferences are a great way to meet people, especially smaller conferences. So if you're like a senior student, if you have a chance to go to a Keystone or a Gordon Research Conference, those are great ways to spend like quality time with other PIs, maybe future postdoc mentors, but also maybe future collaborators. So I think yeah, for me, that was kind of the best way for me to like find out additional mentors.
Yeah, I really agree with that. I also think at your home institution, like when I was a PhD student, I had faculty, for example, like a physicist who studied fluids or a soft matter physicist who I like sought out their expertise. And I did that completely independently of my PI. You know, I contacted them, set up meetings. The faculty are really happy and they love to see when grad students have relationship. So if a grad student came to me and was like, from, you know, hey, I want to chat about my project, that would be honored. The top priority for me. So I mean, don't wait for your like PI to set up all your connections. Like, take the initiative. That's what you're going to, if you do go towards academics, that's what you're going to have to do for your career. Find out people about the expertise that you need, build a connection with them, make them like you. I don't know, that's part of what we do in a way. So, you know, I would encourage you to take the initiative.
Yeah, I mean, I completely agree. And I think that the sooner you start, the better. Because I mean, most of the stories that you will hear is that, oh yeah, my PI is great, you know, and everything is fantastic, but sometimes that's not the case. And, you know, you may need to reach out to some other faculty members or, I mean, whoever, anybody can really be a mentor. And I think the more mentors that you have for both technical aspects and then career development aspects, I think is great. And the sooner you start, the better. And then, you know, programs like this, like the interfaces program, where, you know, all of a sudden, hey, you have a co-mentor, you know, I mean, talk to your co-mentor. I mean, most of the times people have co-mentors or committee members that they, you know, never talk to them again. And, you know, it's not, we tend to see as students, our co-mentors as examiners, and we should see them more as our collaborators and our, you know, I mean, obviously it depends on the person and it depends on whoever you're reaching out to, but some people are going to be very receptive to that type of interaction, and they're going to help you and they're going to give you great advice. So I think it's a great thing to do, and the more mentors that you have.
Yeah, I still speak to my thesis committee members for my PhD, so they're still mentors of mine. There's no rule that says that you can't see thesis committee members outside of your exam, so I would say like maybe a quarter of the students who I sit on their exams, I see them outside of their meetings. So I think that I'm very happy to meet with like anyone I'm staying on their thesis committee outside of the exam. So I encourage that.
If there is one thing you guys could change about your job as an academic, what would that be?
More administrative support. Like, yeah, I think that is the amount of administrative work.
Yeah.
You know, different aspects of teaching and research.
Yeah, fundraising is you know, it's rewarding, but it's also very difficult. So, you know, I have a love-hate relationship with writing grants. It's really fun to put an idea together and actually see it through, but at the same time, you know, you write a lot of grants. So, I guess I like grant writing. I just wish I didn't have to do as much grant writing. I guess that's how I would turn it.
Yeah, I would totally agree with that. I mean, and I guess at the postdoc level, you're still fully enjoying your academic work and doing what you want and what you like. And it's kind of a great position because you're no longer a student, so you don't have that, you know, above your head, like, oh, I need to graduate, I need to do this. I mean, it's like if you're doing a postdoc, you're doing it because you want to.
Yeah.
Most of the time. But I think, I mean, obviously, postdocs are underpaid. So that's one thing that could be better. But and then the other thing would would relate to this job insecurity, right? And that even at the level of assistant professor, I mean, you need to get grants, you need to get papers out, you're on the tenure clock. So I think that's probably one of the worst parts of the academic jobs, that it seems like that it never ends. This fight, you know, it never really ends. And it is stressful. It is stressful. But it comes with few positions that are now available these times, and yeah, it is what it is right now.
Yeah. Should we go to the audience since we're coming short on time? Does anybody have any questions for? Yeah, Joseph.
Just had one main question, but then I just want to follow up with Professor Moody on your response about like administration. I guess like what does that exactly mean? Is that like when you're teaching, you don't want to deal with all the students asking for like points back and stuff like that, or like having to write the exams and for research? What does that look like?
Yeah, I'm talking about very mundane things, submitting receipts for reimbursement, having students trying to get different exemptions. It just all scales. It scales linearly with the number of students in your class and the amount of research that you're doing in your lab. So I think it's just our committee, like we do things beyond just teaching and research, like we do service, committee service, and that's oftentimes very.
Yeah, I guess the question is like, what's the reason that most labs don't have like someone to be able to help the professors in that lab?
Because someone needs to fund that, and when you get a research grant, that's to fund the research. So it's hard to identify the pool. Like NIH, NSF, they're not funding your admins. They can't. You know, they're funding the research project. I mean, there's some ways you can do things, but that's the major reason. And also because research grants, quite frankly, aren't that large. So if you're deciding whether to hire a fantastic postdoc or you're hiring someone, an administrator, you oftentimes postdoc can do science, but they fall into a trap.
I guess the main question that I had for all three of you is the work-life balance, and maybe quantitatively, like how many hours of work a week you work, and what it looks like.
I think the wonderful thing about being a professor is that you can, you're your own boss. Your lab can be as big or small as you want it. So if you want a small lab and minimize the amount of extra stuff, you can. And if you want to do a lot of stuff and have a huge lab and do all the service things, then you can also do that. So I just wanted to caveat the answers with that. There's a lot of like choice. And then let's see, work-life balance. I would say on a typical day, something like 10 hours a day. I often can take weekends if I feel like it, because I mean, at the end of the day, you're only like beholden to yourself. I mean, for me, a lot of my is like, I gotta pay my students, so I gotta that's like kind of what gets like the like I have to do this, because like if I don't fundraise, my students don't have research dollars to do the research and also graduate. So that really gives me that's like my morning coffee is that. So I would say that's kind of typical for me. It goes down a little bit for summer when I'm not teaching. So teaching just takes a lot of time. So that just adds on. So that's kind of typical for me, that I'll work maybe half my weekends, and then realistically, but I think it just depends. Like I know people who are very, very different, that their schedule is very different from that. So.
Yeah, I think as a faculty member, your work and your life is kind of blends together in a bit. I mean, it's your research program, so you're kind of, as you said, putting in as much as you feel that is necessary, but it's not someone's telling you to do something. No one's telling you to do anything, actually. So, I mean, it's almost like the vacation where you have unlimited vacation, but then you don't take any vacation. So maybe it's, but, you know, I think people that work really hard at academics generally do it because they really want to.
Yeah, I think there's a lot of personal choice. I mean, obviously, like the short answer would be, well, it ain't pretty, in the sense of you're gonna have to work quite a bit because of the competitive nature of the positions that you're gonna be applying to. So you definitely want to show that that your work produced certain output. But at the same time, there's a lot of personal choice, and even within the same lab, you're going to see people who are going to work themselves to death, and people who are going to take it a little easier and say, hey, I'm in here, I'm not going to be here for 2 months, I'm going to be here for 2 years or whatever. So I definitely want to have a more steady path and kind of preserve my well-being and my mental health. And I think with COVID, all of that became more apparent because we've all been there. We've all been like working from home when you don't really want it to. And it kind of gets to you. I mean, when I started my postdoc, for example, I would say the first 6 months, I couldn't even get into the lab. So I couldn't really get started. I couldn't get training in a lot of things that I need to get trained on. All my interaction with lab members was over Zoom. I didn't meet my PI until month number 4, where we went on a hike. So you definitely wanna also pay attention to your well-being and to your mental health and say, well, and work-life balance means different things for different people. So I think as long as you are okay with what you're doing, then some people really enjoy working 12, 14 hours a day and they're happy doing that. I mean, that's great. But if you're not in that pool of people, then don't overdo it because eventually you'll pay for it.
We'll take what yours as the last question. We're running out of time. So yeah, 10 minutes. Oh, we have 2 minutes? Okay. Yeah, go ahead.
I have a general question. So, just when as a PhD student, when we're looking at positions, say we should maybe focus on the publication record of that professor to see, tenure go, or maybe looking at the institution to see what sort of resources they have, what we could leverage on that, or other things that we should look at. What sort of positions there's not.
I think a good thing to check is, did their postdocs go into faculty positions? If it's a newer PI, you'll have less data, so you'll just want to do more research in terms of talking to the people in that lab, publication records, all that kind of stuff. But if you're going to an established lab, you can see their track record of putting people into faculty positions, so that is helpful because it requires a lot from the advisors, like their support to get you to do the faculty applications, including their letters and like how much they're going to advocate for you. So I found that to be pretty useful. Besides all the other things, right, like research environment, those are also important.
Also, you might want to consider where you're living because, like, as a postdoc, you can choose. You have a lot of choice. When you go on the faculty job market, you're going to have much less choice, and you have to really consider like what is going to be the best place to have your research program flourish. I think, you know, when you're looking for a postdoc, you can, like grad school, you can have, you know, those geographic choices. You're going to be twice for the last time. So I don't know, no one ever said that, but I would also play that factor.
Yeah, I completely agree with both of you. And like, for example, when it comes to life choices, I mean, I was telling you before that I spent a year at Yale working with this BI. I applied to do my PhD there, and I got admission, and I still came to UC San Diego, mainly for two reasons. The first reason is that I wanted to do more bio stuff, so it was kind of in my career plan. And the second reason was because, well, I lived in Haven for a little over a year, and I didn't really see myself spending there 5 more years or 6 more years. So you definitely want to consider these sort of things. And then when it comes to choosing a lab, choosing a school, obviously, I mean, everybody likes big names, and that's great. But at the same time, I don't know, like I came to UCSD, for example, and to discover that there were amazing labs at UCSD, and I didn't know much about how important the bioengineering program at UCSD was, and things like that. So names don't mean everything, and then you definitely have to try and choose the lab that aligns better with whatever plan it is that you have at that point. And then, I mean, talk to people who are currently in the lab, talk to people who have left the lab already and might be more honest with you. And listen to your gut also. Because sometimes we do have gut feelings, we don't listen to them, and then we realize that we should have listened to that. And a lot of times, I mean, when you talk to people, you're gonna get really valuable information, and they will tell you, you know, this is a great environment, the lab culture is great, the PI is very generous, you know, when it comes to letters of recommendation or even treating people equally, not having like favorites, things like that. I think all of these things are quite important.
You know, one of the most fundamental needs of outcomes for particularly first order publication, particularly these days in our field, impact. How long is it going to take you to write that first order publication? What's the chances that it's going to appear in a journal? There is a huge variety. It's not always the case that the big names get the person you want to be with. Maybe all the papers of postdocs are all in self-vaccination. But how many of those postdocs get those papers and how long does it take? I always remember there was a postdoc in the lab of my postdoc mentor, who is a very famous, and pretty much they only wrote papers themselves. And this guy, Timo, was funny because he was just like, all I want to do is write a paper with cell and die. And he spent 6 years developing a mouse model. And the month that he developed the mouse, someone published the same mouse itself. He went back to Finland without it, but they published it in paper. So I would remember that. It's like, this is a famous lab, but doesn't guarantee.
I will mention regard to that and timeline, if you are interested in going academics, like the clock starts ticking like when you start your postdoc, in a sense. So when I mean by that is, don't like, I see PhD students who like start a postdoc early in their PhD lab. Don't do that because it actually excludes you from a lot of postdoctoral fellowships that start once you take a postdoc position. So I would advise not to do that. And I would advise like actually spending a little extra time. I know NIH doesn't like that for training grants, but finishing your papers in your PhD and getting like, if you have, multiple PhD papers, that's really good. And then you get your, your first author postdoc paper and go in the job market right then.
And take a really good long vacation between your PhD and postdoc. The last chance, or one of the last chances you'll have. Like I took 2 months off and went to France and whatever. And it's like, it's not on my CV. No one knew that I did that. Okay.
But yeah, I mean, kind of to build up on both comments, like a lot of it is also luck, in the sense that, yeah, being on big labs doesn't guarantee you anything. And for example, I graduated in February 2020. So you can imagine what vacation I took. So when I was ready to do this sort of thing, then a pandemic hits. So there's a number of things that you can control and that you have to, be flexible about. And kind of take it with a grain of salt and keep being optimistic and, keep pushing and.
Also know what your field expects from your postdoctoral work, because that can actually be dramatically different depending on discipline, how much postdoc papers and what journals are expected to get faculty jobs. So when you're actually, even as grad students, you can see people coming in for interviews for open positions in CSD, and you can see what their resume is, and that should give you a sense of what the expectation is. And that can be very different depending on.
More questions from the audience?
I guess one last question. This maybe this is personal in a sense, but as you become like a PI and as a faculty member, you're further and further away from the actual hands-on science. And so do you miss that? Like, how do you deal with that? Or is it more exciting that you just come up with these ideas and tell people to go do it? What's been your experience there?
I'm experimental at heart. I love doing experiments. I still do experiments with my students. I know also faculty who 30 to 40 years into the lab, they still have their own bench. So it is really like whatever you want to do, because there's no one really telling you what to do. So that's how I run it. And I probably will still do experiments for quite a while.
Yeah, I would love to actually have more time to do experiments myself, but I also know people who think the exact opposite.
Still doing experiments. No, but I think, it is exciting when you start having ideas and then you realize, well, I may not be the one working these ideas, and these are going to be ideas that potential students or other people are going to work, and to me that is also very exciting. So.
Awesome. Well, let's give the panelists a round of applause. Shot after Professor Lindsay's final keynote.
Mostly for the recording.
Yeah.
Just wanted to introduce you guys to Stephanie Lindsay. She's a Whitaker International postdoctor scholar and also an NIH postdoctor fellow in the past. And she completed that postdoctor fellowship in France. And her research has been focused on cardiovascular modeling in both native vessels and tissue engineered constructs and its association with hemodynamics. She relies heavily on numerical simulations to close the gap between experimental and clinical data. And she received her BS at Washington University. She did research in on the mechanics of morphogenesis lab. And she received her master's and PhD in biomedical engineering at Cornell University in the cardiovascular development bioengineering lab. And so, yeah, just want to introduce Stephanie Lindsay to you guys.
Thank you. So I just arrived here at UCSD in July, so it's my first year. So you can also ask me some of those panel questions if you want after the talk. Happy to answer. So yeah, so this is just sharing a brief window into what we're doing in my lab and what I've done in the past. So this talk's entitled Engineering Redundant Solutions for Congenital Heart Defects, which is the main focus of our lab. So this is some background. So your normal healthy human heart separates oxygenated and deoxygenated flow, and it pumps oxygenated rich flow to the rest of your body. And so when development doesn't proceed as planned, that's when congenital heart defects arise. And so shown here are some of the more severe congenital heart defects of the outflow tract. So the outflow tract consists of your pulmonary artery and your aorta. And so here we have persistent practice arteriosus, where they didn't fully divide. There's also this ventricular septal defect, so just a hole between the left and right ventricle, transposition of the great arteries, where the aorta and the pulmonary artery are switched. And then this is hypoplastic left heart syndrome, where the left ventricle is severely underdeveloped, almost to the point of being non-existent. And it comes with this characteristic narrowing of the aorta. And so this is the most severe congenital heart defect, falls under single ventricle abnormalities. And this up here is taken as a section post-mortem, an autopsy showing you this really thin narrowing in the aorta. And so I look at this and I wonder, what is the role that blood flow has in the creation of these defects? And so that's kind of what we focus on. And so traditionally, this has been studied from the point of view of developmental biology. And so with the advent of all these new sequencing and technologies, we've identified many genes that are associated with congenital heart defects. And the problem is that correlation doesn't always equal causation. And so here we're just looking at a small portion of chromosome 22 that's been deleted, and it's associated with this larger array of congenital heart defects, a lot of them involving outflow tract, and including transmission of the great arteries that we saw on the first slide as well. And so what I do is we work to come in with our engineering toolset and see how we can use basically really highly specified technology to create really specific experiments and integrate this with computational modeling in order to better understand disease formation and generate redundant solutions. And so as one of our thrusts, we look at this from a developmental biology point of view. And this is really because in order to address problems at their root, we really first have to understand how they're formed. And so we look back into how the heart was formed. And so this starts with the specifications of myocardial cells, the formation of the cardiac crescent. Then we have a single heart tube, which has an endoendothelial sheath and an outer muscular sheath as well. It loops once, it loops again, and then it septates into 4 chambers. And actually, if we perturb this flow before it reaches 4 chambers, then we can create a number of congenital heart defects. And so these are just a body of animal simulations
where they do both genetic and mechanical perturbations that ultimately change flow structures and change how the heart develops when it's four chambered. So just some of these, we have left atrial ligation, where sutures slide over the left atria. It's pulled tightly enough to arrest flow. And this actually can recapitulate an animal model of hypoplastic left heart syndrome, where the left ventricle was severely underdeveloped. And if you go in a few days later and you put a clip on the right atrium, then you can rescue this. And so there's different clippings and ligation models, also from a genetic perspective. So here, this PIX2 mutant, so PIX2 is responsible for the spiraling of the outflow tract, which the outflow tract is pulmonary artery in the aorta. And so it starts off in its single form. And so this spiraling was prevented in the PIX2 mutant, and it led to a number of congenital heart defects. And then at the bottom here, we just have the deletion of one BNPR1 receptor in neuropress cells, and that led to persistent pancreas arteriosus. And so if we couple these experimental observations with computational simulations, then we can better understand why what we're seeing experimentally happens. And so just at the top here is just computational fluid dynamics simulation of the left atrial ligation. And so the question is always, what comes first? The structural defect or the fluid flow, what's causing what? And so here they perform this simulation to see if their hypothesis that the immediate flow effects by tying that ligation, if those were responsible for the hypoplastic cellular response that's to come later. And they actually found that it was. So there were areas of really low wall shear stress, and those areas were the areas that were going to become hypoplastic. And so they could follow these changes. And you can also do this on a more tissue level, just by tracking tissue deformations. So here at the bottom, this is from the chick embryo, and really just being able to track tissue deformations and create a shear strain field and understand how the embryo goes from being a flat, basically just a flat sheet into its first 3D movement, and that will initiate cardiac formation as well. So going back to how we study abnormal cardiac development, and this is really using highly specified technology and using that to create these targeted experiments. So I did my PhD at Cornell, which is really big on imaging. And so at Cornell is where they actually came up with two-photon microscopy. And so I was in a soft tissue biomechanics lab, and then we collaborated with this nonlinear microscopy lab. And so what we use from there are two-photon microscopy and femtosecond laser ablations. So two-photon microscopy is just your imaging laser, and so it requires two photons arriving at the same time. And so it's dependent on the square of the excitation instead of linear. So basically everything that you see here in this really tiny volume is what can possibly be damaged by the laser. Versus with regular linear microscopy, everything that's fluorescent can possibly be damaged. And so this is what's going to allow us to image deep into the tissue. And so we're using the chick embryo as our animal model. And then we're going to combine this imaging, which allows us to just image anywhere within the heart and the entire vasculature. A great thing about the chick embryo is that it's all exposed. And then we combine this with these femtosecond laser ablations. So they're really brief, very powerful flashes of light that are contained in this femtoliter size volume, which we can align perfectly within the two-photon microscopy beam. And then we're able to perform these occlusion experiments. And so basically, at the stages that we're performing occlusions, the chick doesn't have clotting factors. So normally, if you are going to occlude something and turbine and you can actually form a clot, you actually disrupt the vessel wall, and you initiate this cascade of events. But what we're doing here is we're focusing the femtosecond pulses just in the center of the vessel, and we're creating these cavitation bubbles, these femtoliter-sized cavitation bubbles that we can then grow, and we can block blood cells, so you can actually see the cells traveling through these vessels. Then we block the cells and we start lysing them with the ablation laser. So we lyse the cells,
they become sticky, so they form this clot-like structure, and that's how we were able to occlude different vessels in the developing heart. And so the vessels we chose to occlude are the precursors to the great vessels, which is the aortic arch, the pulmonary artery, and all of the branches off of the aorta. So we occluded these before they're in their final stages, and this led to a number of defects. So this is just 24 hours after the insult. So basically all flow that exits the heart has to go through these pharyngeal arteries, is what they're called. So we occluded it, and the embryo tried really hard, whatever it could do, to keep a connection with this dorso-aorta large vessel that's running along the back of the embryo. And so basically, normally the vessel gets to be quite large and is directly in the middle, but it would just have this slither, anything it could do to keep a connection from the heart to this dorso-aorta. And so what we do is then we integrate these experimental findings with computational modeling. So we have our experimental geometry, then we send it to nano-CT to get really high fidelity scans. We're able to get a 3D rendering, and from the 3D renderings, we can perform computational fluid dynamics and have these maps of all the forces taking place. So mainly the forces that we want to track are wall shear stress and pressure, and see how those are affecting development and how those have changed. And so what we can do also, harnessing on the power of these computational simulations, is we take our occlusion geometry, and we can also, from controls that weren't occluded, we can perform these in silico occlusions. So we can do different degrees of full and partial occlusions. And we can say, theoretically, if the geometry couldn't remodel, what happens to the physics? So like, how would the flow, how would the pressure change? How would the wall shear stress change? And exactly what would happen? And then you can compare that with known growth adaptation mechanisms and compare those with the experimental findings to kind of validate and come up with different growth and remodeling paradigms. So basically, using a combination of these in silico and comparing them to what actually happened experimentally, we could distinguish between the growth and remodeling mechanisms. And we were able to propose this two-phase vessel adaptation mechanism. And so all of this was possible from the experimental, not just so running computational fluid dynamics simulations and reconstructions of the experimental geometries and comparing those within silico simulations. So where we basically gave it an inclusion or exclusion. So just from this first developmental biology thrust, by identifying the root causation, we can better understand disease formation. And this is really a launching pad for regenerative solutions. So the goal of my lab with this thrust is really to be able to design in utero surgeries so that we can reverse these defects if we really understand how they form, where exactly they're forming, and a timeline of events. So that's the goal there. And then we can also try and optimize current procedures as well. So, it takes a little bit of time to go from our trip to the clinic, even though there's so much potential for what we can change. So we can also, as another thrust, just optimize current procedures. So if we go back to the cycloplastic heart syndrome, where we have the left ventricle that's severely underdeveloped, this actually requires 3 open heart surgeries to create a palliative solution. So palliative because we can't regrow the left ventricle. But what we can do, so this used to be a death sentence right before the 90s when they came up with this series of surgeries. But what we can do is, give the child and will become an adult, a much longer life and a quality of life. And so the third of these surgeries is the Fontan procedure. And so the problem with the hypoelastic left heart syndrome is separating of oxygenated and deoxygenated blood and getting oxygenated blood pumped to the rest of your body. And so basically through these 3 open heart surgeries, we're recreating a circuit that will allow for the separation of oxygenated and deoxygenated blood. And the third procedure, a synthetic graft is generally inserted, and this will close the circuit and with the TCPC junction. And so it's generally this foreign material and actually leads to a number of complications in adults. So congenital heart patients with congenital heart defects, they're the largest percentage of the population that see the cardiologist as an adult, because you're correcting, they're so small, and then, for example, this graft or a lot of the corrections that we can do, they can't grow with the patient. So a huge problem is like this graft can't grow with the patient. And so basically, this is in collaboration with the Brewer Lab from Nationwide Children's Hospital and the Humphrey Lab from Yale University. But the Brewer Lab tried to come up with a solution with a tissue engineered vascular graft that's actually capable of growing with the patient. And so they underwent these clinical trials, and they showed that the graft can actually grow with the patient, and there was no graft-related mortality, but there was actually stenosis, or this narrowing of the graft, that was seen in every patient. And so even though this didn't kill anyone, it didn't really affect their quality of life. It's really hard to convince anyone to adopt this new surgery if you have to go in and do angioplasty. So go in with a balloon and they're going to have another intervention. So they're signing up for another intervention. And so the Brewer Lab came to my postdoc lab at Stanford, the Marsden Lab. And they're like, what can you do with computational simulations to help us understand how we can optimize this and optimize the design of tissue engineered vascular graft and understand why this stenosis is forming. And so what we did is we did a parallel study. We simulated in the patients, and then we also used this ovine surrogate model because we could perform a lot more invasive measurements. So in the ovine surrogate model, we call it an interposition model. So basically, instead of attaching the graft to the TCPC connection, it's attached to the right atrium. So it's still sitting in the inferior vena cava, so it gets the same load volume, the same type of flow, and so this is performed in sheep. And so these are just reconstructions of 1 week post-implantation and 6 weeks, and already at 6 weeks, the stenosis formed in all of the sheep. So it's a good model to study. We're seeing the same thing we're seeing with the patients. And so basically, this is how the stenosis is perceived at the level of the fluid. So it's perceived as this really tight narrowing. So this is intravascular ultrasound here, and this is histology. And so you can see that it's not like the graft is being sunk in. There's actually this complicated growth. So it's inward remodeling and intramural growth that's occurring at the level of the scaffold. And so basically, we do our simulations. So we get our geometry from MRIs, we get our flow curves also from the MRIs. We match pressure drop from the cath lab, from angio, and we also match the deformation data and get thickness from IVUS. So this is as realistic as we can be. And so here is what they look like for 1 week, 6 weeks, and 1 year post-implantation. So at 1 week, there's still the scaffold, which you'll see in this purple kind of color. So you can see it in 1 week, you can still see it in 6 weeks, and then this green is collagen building in. So basically the scaffold breaks down, and then you have a neotissue formation. So, and then it should assume some kind of mature vascular phenotype by 1 year, and that's what we're seeing. The red is more mature collagen that we're seeing over here. And then here is how the fluid is affected for all of those. Okay. So this is just an area and thickness breakdown. So something that was really curious is that the stenosis occurred at exactly the same area for all of the sheep. So there's 8 sheep in here, and it always occurred at 0.8 of the way from the inferior vena cava towards the aorta. And so you can see that there is this narrowing, but then there's this huge thickness that's occurring at this narrowing. So it's like encroaching on this narrowing. And then here's how the hemodynamic responds, exactly as you expect. So if you're creating this huge narrowing, there's going to be this huge pressure drop. So very large pressure that we're seeing at the 6 weeks. By 1 year, it's actually approaching native again. And so you see the wall shear stress profiles as well. So very bumpy for the 6 weeks. It's elevated because there's a larger flow volume at the 1 year, but they're still relatively stable along the arch. So we're looking along the tissue engineered vascular graft length. And then this is what's experienced by the cells and the tissue itself. So that's experiencing the transmural pressure or Cauchy stress is what we can see there, as well as its deformation can also be described by the screen strain. And so what we can do to further understand what's going on a tissue level is plug this into a growth and remodeling framework. So this was actually performed by the Humphrey Lab, Jason Zachan at the Humphrey Lab. And this is a constrained mixture growth model. So basically for the constrained mixture growth model, we can understand growth and remodeling as part of its components. So here there's going to be an immune response and there's going to be a mechanical mediated response. And we're actually tracking the addition or removal of different cellular populations. And so you'll have this initial scaffold, which is what we're implanting. And then this will start to degrade. And basically we'll have the immune and the mechanically mediated stimuli that are deposited at stretch. And then this causes it to deform into the current configuration. And we can track all of this just by multiplying all these deformation gradients. And basically we can track how all of the constituents are changing at any point in time. And so we had to tune the GNR framework to match the diameter and the wall shear stress measurements. And so in white, you'll see what we measured. In black is the growth and remodeling framework. So up at the top is us tuning. And then down at the bottom, we perform these inflation, pressure inflation to actually see how well the GNR framework matched the experimental results. So once again, the experimental is in white, and then in black is the what it's being predicted by the framework. And so compliance, you can think as kind of a measure of the stiffness of the graft. And so the prediction was very well. And what you can see here is that at 6 weeks, when it's the thickest, this is actually where the immune response is peak. And then the immune response will start to wane out somewhere around 6 months. And then as we go on, it'll just be a mechanically mediated response that's actually responding here. And so that's what we know from how the GNR model, how we were able to get the GNR model to match the experimental results. And so we can further verify this timeline by taking measurements from our computational fluid dynamics simulations and plugging them in to idealize wall shear stress and Cauchy stress or hoop stress, so from the transmural stress acting on the wall. And so basically, if this was purely a mechanically related response that the tissue engineered vascular graft and the neo tissue, so the new vessel wall was responding to, then it would be following this dashed line if it was purely mechanical. And so the white is actual measured values, so for thickness and for radius. And so basically the difference between the dashed line and these white markers is the immune response. And so this further validates exactly what we saw with the GNR framework, that the immune response is at its peak here, and then it'll wane off. And so become much more mechanically led, closer to the 1 year mark. So for the conclusions for this second palliative care optimization for us is that numerical simulations really help us to understand the inner workings and disease progression, and we can optimize current solutions. So actually, from these models, what we're able to predict and show was that the stenosis would spontaneously reverse itself. And so basically, we showed that stenosis spontaneously reversed itself, and also by doing a closed loop model where we could study the effect on all of the pressure and flow curves for the body, there was actually no effect. So there was no adverse effect if it went to 80%. So they were doing these angioplasties really quick as soon as they saw the stenosis. So they're like, oh, we have to fix this. If we were modeling, we're able to show them that like, actually this will spontaneously reverse itself. And unless the stenosis gets to be like 97%, there's actually like no effect felt in the body, so you don't have to panic. We're working on changing the materials of the graft and helping them optimize the design so maybe the stenosis won't form in the first place. But even with these first running through, we were able to Actually, there's a new clinical trial going on now, and they're using new parameters based on these simulations. So with that, I'll thank my funding sources, and I can take your questions.
I had a question about the, I guess, the synthetic stuff that you put in the heart and the graft, and like what it's made of and how you put it in and how you came up with it.
So for the tissue engineered vascular graft, it actually has an outer PCLA sponge and then an inner mesh that will actually give it its robustness. And so the inner mesh is actually what we're playing with different. So for these simulations, it was all PGA, and they're also doing some simulations where it's PLLA, so poly L-lactic acid. And so these are all from the Brewer lab, it's a tissue engineering lab, and he's actually a cardiothoracic surgeon. And so he came up with this idea from all of the Fontan procedures and all of the complications. So basically, patients have to go on anticoagulants. There's this thing where they'll do an oversizing of the graft, so they'll try to make it so to give like the patient a little bit of time to grow, you know, a little bit of room to grow. But it's just really complicated, and the ideal thing is to have a graft that can grow. And this is not the only application. So this was a very obvious application because the graft is used in every single Fontan procedure, and it's, I mean, it gives a lot of hope to the patients. So a lot of people will enroll in the clinical trial. But this can be used like anytime there's a graft. So it's able to actually grow with the patient. No anticoagulants and other things.
I would say how incredibly exciting it is to see this particular class of model already kind of making its way into, you know, informing clinical practice and surgery, because modeling biomechanics with flows and forces and stuff and using it in surgery, that's not new, but making models that actually predict the model based on the biomechanics. That is much newer. Really just impressive to see that you're already at the point where you're making predictions that are, you know, enabling surgeons to change their procedures. It's really exciting. And that question is how, you know, the rules that those growth and remodeling models use, so they're empirical. So they're sort of based on observation and they're ultimately based on hypothesis.
Yeah.
But we know that they're underlying their mechanisms, right? They're mechanosensitive signaling mechanisms and so forth. What do you think the prospect is for coupling those together so that we could have models that not only predicted the phenotypes, but actually predicted the mechanisms, so that you could perhaps also superimpose or alternatively use medical.
Yeah, so actually the Humphrey lab has already started doing that. So coupling more agent-based molecular pathway models with this. And it's relatively quick and lower cost to do those simulations. So it's not a hard thing to couple. What we've been working on coupling is the GNR directly with the fluid structure interaction or computational fluid dynamics simulations, which is like much more costly. But actually just coupling like the GNR itself, that it's definitely feasible, and that we're already doing that. So it's nice.
Very exciting.
And just another comment is that so the first time that the Humphrey lab saw spontaneous reversal of the stenosis was actually in their simulation of a mouse model. So they started, sheep experiments are very expensive, and all this work in order to be able to do all the sheep.
but the large animal studies are more clinically translational and accepted, by NIH and everyone. But they first saw it in the mouse model. And so it's just like a lot of times people ask like, oh, you're using animal models, like how translational is what you're doing, but it exactly translated from the mouse to the sheep to the human. So that's also something cool.
Perhaps I just didn't catch this part, but I had a question about the stenosis. So of all the responses that possibly happen when you put in, thick tissue, why does the wall choose to grow so thick?
Yeah, I mean, it's largely like the immune response, right? So it's like this foreign body response is activated whenever you put in something. So even though like these tissue engineered vascular grafts are actually seeded with patient cells, but even so, we get this foreign body response as soon as it's implanted, and so that's actually just part of the immune response. Is this inward remodeling?
I guess I'm surprised that the immune response is to thicken the wall instead of like, for instance, grow new blood vessels to come out and find an alternate route or something.
Yeah, I mean, it's so complicated, kind of thing. I mean, because like the first thing, what we wanted to do is to like break down and degrade the scaffold, right? So growing new vessels is like so many different steps, like kind of thing, and like you still have to have some kind of cellular growth and all of these things. And so this is just the cellular growth that we're seeing. This last one's dying. Okay.
One last question.
Yeah.
Sure. In terms of, you saw the stenosis forming out like the kind of normal length of like 0.8, right? And so did you notice that was like a critical like stress or strain, or was it like turbulent flow starting to flow there? Or do you know what was special about that?
Like why it was like, so it's like, it's really close to the distal anastomosis, right? So like where they're actually connecting the tissue engineered vascular graft and the native vessel. So we're like, okay, can we change something about like the sutures or like anything? And we actually haven't figured out exactly like why it's still at that 0.8, but it's something that we're like really looking at in terms of tissue properties and everything too. Yeah.
So when you see this reversal, about how long does it take to reverse, and is it also due to like some type of immune response?
So it takes about so the scales are different in human and so in human it's actually 6 months where we see like the stenosis form. So it's about a year and a half when we see the reversals, like a year after that. And so yeah, so the increase in pressure and everything too also helps with the degrading and the reversal of the stenosis. So it's kind of like helping itself. And so no, the immune response has waned by the time it's like actually reversing, which is good. So we don't have to like keep countering the immune response. But then we can just, now it's like regular growth and remodeling adaptation mechanisms are applying. And so these heightened forces are actually helping.
Thanks so much.
You're welcome. Okay.
Oh, wait, is that some award?
Stephanie is the
I guess, yeah. I'll
I don't want to slow it.
There's actually a tie. All of you guys did between Christian and me. So these are all winners. So.
Now is that? Okay. Well, so we still have a mixer networking. It looks like there's got food bowing outside, so I hope everyone, I know Dr. Lindsay has to go too, but I would like to, first of all, just thank you all for coming along. And it makes such a big difference to do this in person. And really special thanks to Kelly and Irene for all the work that you did. We might be using the bags, you know, that we have to like take all the extra food with me we can go. And thank you again, all of the organizers and all of the judges and our alumni visitors and our faculty speakers. This was really, really great. And we really appreciate hearing from the faculty. Thank you, Stephanie, for coming. I know you have to teach, and it's the unique part about this program that we have such strong interdisciplinary ties across the different programs and such interest. You know, there's only a certain kind of student that wants to be in a program like this. As we were talking about in the discussion, just being a PhD student is a daunting thing. And, you know, I always said actually that when students come to me and ask about graduate school, I always say, for years I've always said I would never talk anyone into doing a PhD. You know, you can't be talked into doing a PhD. It's kind of like being talked into getting married or something. It's probably not a good idea. You know, you really have to want to do it. But you know, that takes a lot just to take that step. To take the extra step of saying, okay, now I'm going to join a program where I'm taking classes and doing projects and attending symposia and retreats and lunch seminars with students from lots of different programs where I really have to sort of work at understanding what they're doing and finding common interests. That's really kind of special. And so I really appreciate that in organizing the symposium, you kind of embraced all of that, that interfaces goodness that we have. So, so thank you all. Let's go outside. Hopefully it's not too cold yet and we can enjoy the view and the reception. Yeah, that, your badge. My badge seems like stick that way out. Thank you.